Hey, welcome to our 2020 Translational Science Workshop focused on taking MPMI discoveries to the field. Today's workshop is hosted by the International Society for Molecular Plant-Microbe Interactions with generous sponsorship from Microbes Biosciences. My name is Mary Beth Mudgett. I'm the current president of IS-MPMI and a professor at Stanford University. I welcome you all to this event. We are joined by members of our IS-MPMI Society, members of the American Society of Phytopathology, and colleagues with shared interest in research aimed to improve plant health worldwide. The goal of this forum is to directly engage with leading scientists working at the forefront of fundamental research in different areas of MPMI who are translating their novel discoveries to the field with the ultimate goal to develop disease-resistant crops that will positively impact global food security. Today, we will hear from 4 distinguished professors whose science and actions are leading the way. We welcome Jan Leach from the Colorado State University, Brian Staskowitz from the University of California at Berkeley, Pamela Ronald from the University of California at Davis, and Brian Stephenson from the University of Minnesota. Throughout this forum, our speakers will highlight technical advances, progress in their fields, and share challenging issues for bridging the foundation of research and translational gap. And as a community, we can capitalize on this in our expertise of IS-MPMI and APS communities to exchange ideas and strategies to bridge this gap to promote plant health around the world. I have an honor to moderate this forum with 2 wonderful colleagues, Jean Harris and Dennis Halterman. Jean is a professor at the University of Vermont and is the current editor-in-chief of the MPMI Journal. Jean has revitalized the journal and its activities and hosting MPMI virtual talks during this pandemic, which has been vital to bring exciting findings that are published in MPMI to both the data for us and also to meet the scientists in our community. Dennis is a research scientist at the USDA in Madison, Wisconsin, and the editor-in-chief of IS-MPMI Interactions. Dennis is an active member of both societies and has been an essential leader in activities aimed to maintain interactions between IS-MPMI members throughout the year. Before we begin, I'd like to give a brief overview of the program today. The workshop will be recorded and available for viewing in future if you're not able to stay tuned for the entire segment. The order of the talks will be shown. Can we have the first slide? Can we, thank you, Brian, or Nelson. We will begin with Jan Leach. Each talk will be 30 minutes, followed by 15 minutes of questions and answer. Then we will have Brian Staskowitz, followed by our sponsor, Brian Push. We'll take a brief break, and then we will finish up the latter part with Pamela Ronald, and then Brian Stephenson closing out with some final remarks and questions and answer period. A little bit of housekeeping notes for today. We will be using the Q&A function to take questions at the end of each talk. Please go ahead and enter your questions at any time. We have reserved 15 minutes for this, and we look forward to hearing from you. Our webinar is being run by scientific societies, and Nelson will be assisting. So if you have any issues, please contact Nelson via the chat. Okay, let's get started. Dennis, my co-moderator, will you please introduce our first speaker?
Thanks, Mary Beth. Jan Leach is the Associate Dean for Research in the College of Agricultural Sciences and a University Distinguished Professor at Colorado State University. Jan's research focuses on understanding the molecular basis of durable disease resistance, particularly in rice pathogen interactions, and how resistance is affected by climate change. Her research program also includes studying complex interactions that occur between plants, insects, and the microbiome. Jan has devoted her career to understanding how to stabilize plant disease resistance to reduce crop losses. The impact of her work is increased by her ability to collaborate with international scientists in diverse fields. Jan's work provided the first rational approach to predicting how long disease resistance in a crop would last in the field. This discovery allows crop improvement experts to select the most useful and potentially long-lasting resistance traits prior to committing breeding efforts to introduce them into advanced varieties. Jan's international collaborative team also discovered multiple genes that confer resistance to diverse types of plant pathogens and demonstrated that accumulation of these genes can confer highly effective broad-spectrum resistance in the field. In addition to her distinguished research success, Jan is also an outstanding mentor and a strong advocate for undergraduate research and women in science. This year, Jan received the Award of Distinction from the American Phytopathological Society, the highest honor the society can bestow, and she joined a very select group of only 16 recipients in the society's 112-year history. In her deanship, she has assembled a new generation of molecular plant pathologists at Colorado State, fostering community and collaborations, which are the hallmarks of her research legacy. I'm excited to hear about Jan's translational and transformational work in her talk entitled Uncovering Complex Traits for Broad-Spectrum Disease Resistance.
Thank you, Brian, and welcome to all of you. Good morning, good evening, good afternoon from wherever you are. Are you seeing my screen, Brian? The correct screen?
Yes, we are.
Thank you. Thank you. Well, thank you for the introduction, and I'm very happy to be here today. This is a wonderful webinar, and it touches on an area that's very dear to my heart, which is translation of our MPMI type research into the field. So today I'd like to give you a brief update of some of the research that we've been working on for the past 30 or so years in my laboratory. And the topic we'll touch on today is improving broad spectrum resistance in rice. And some of the, at the end of the presentation, I'll give you a few thoughts on the translation of these findings, some of the lessons that I've learned at least over the years in translating these findings into the field. So the system that I'm going to spend my time on today, which is my favorite system, is rice. And rice is, as you know, a staple food for over half of the world's population. And it's faced with many disease pressures. And these are continuous, always occurring in the field at all times in all places around the world. And most of these are common diseases that we find throughout the world. Our strategy for disease control has largely relied on the incorporation of single resistance genes, and they're very, very effective in most cases. As you can see here in this particular example, you don't need to be a plant pathologist or a breeder to see that this line is resistant and this line is heavily infested with bacterial blight of rice. But unfortunately, these resistances, as we know, are unstable. And while it can take a breeder 10 to 15 years to identify a source of resistance and move it into the host plant, those resistances can break down sometimes as early as 2 years, sometimes 5 years, but that's totally unstable or unsustainable. So we detect virulence in the field a few years later, then the resistance declines in that field. And this is an example of a case in Korea many years back where they released a new source of resistance, and within 2 years that source of resistance was broken down, and you can see the field was totally decimated with bacterial blight. So our long-standing goal has been to underpin the single dominant resistance genes with a broad spectrum durable resistance. And that is, I'm going to use the definition, is defined as long-lasting and effective against multiple pathogens. For a plant pathologist and a breeder, it's the Holy Grail. And the Holy Grail is known to provide happiness and eternal youth. Well, we can't promise that, but we can promise food in maybe not infinite abundance, but more abundance if we reach that goal. So my group has been focused for many years on identifying quantitative traits for disease resistance. And these genes encode the basal resistance that we're so familiar with. QTL-based resistance is controlled by many genes, or it's multigenic, and it's frequently effective against very diverse types of pathogens because it's of the nature of the type of resistance. It's also predicted to be long-lasting because of the diverse mechanisms that are used in QTL-based resistance. But unfortunately, it's difficult for plant breeders to approach QTLs, and historically, because the genes that are responsible are not known, so you can't target them specifically for breeding. You might use markers that are associated with a gene, but if they're not intimately linked to that gene, you can get to the end of a breeding program and you will have no resistance. The resistance is also difficult to breed for because it's partial effects. So, a minor effect QTL may account for only 10% of the resistance observed in the field. And that's very difficult to score for, where single gene resistance, when you screen for a plus minus effect, is it's much simpler. So today I'm going to tell you two stories. One is how we've used a novel and new-ish population called a magic population to help us identify new sources of broad-spectrum resistance for rice bacterial diseases. These two pieces of work have been published, but the key players are Ana Bosa, who was a graduate PhD student, Ali Huerta, who was a postdoc in the lab, and Emily DeLorean, who was a master's student in the lab. But I want to point out that you'll notice there are many authors on these papers because we have had to have, in order for this work to be successful, multiple collaborators from around the world. And you see here Valerie Vider and some of our key collaborators in Mali for the first project. And other researchers from the International Rice Research Institute will appear in the second project, in both projects actually. So our two pathogens for this story are Xanthomonas oryzae, Pathophara oryzicola, which causes bacterial leaf streak of rice. And interestingly, this pathogen, which is very closely related to Xanthomonas oryzae, the bacterial blight pathogen, this pathogen lives in the intercellular spaces. It has a whole different phenotype or pathology than bacterial blight, which lives in the xylem vessels. Now, interestingly, we have many disease resistance genes to control bacterial blight, some of which break down quickly in Asia, but in Africa, we have very few sources of resistance that have been identified and have been moved into African germplasm for bacterial blight. Xanthomonas oryzicola, while it's very similar genetically to bacterial Xanthomonas oryzae, is very interesting in that we can't so far have identified very few single R genes that confer resistance to this pathogen. And so we've relied in the field on quantitative-based resistance to control this pathogen. And again, this pathogen, which is emerging across Asia and Africa as an important pathogen, has very few sources of resistance, especially in African germplasm. So we set out to identify some sources of resistance, and we thought, well, why don't we initially start, instead of looking for single gene resistance to these pathogens, look for quantitative resistance. And so at the time we started this work, the International Rice Research Institute had developed this novel MAGIC population, which is a multi-parent advanced generation intercrossed population. I want you to notice that this is the population planted out at the International Rice Research Institute. And you should notice how even and how similar the germplasm is. And that's because when they selected the parents for this population, which are 8 parents. So you're intercrossing 8 different parents rather than the normal populations we use, which have 2 parents. These parents were elite cultivars. And that's important because for us, when we get down to the end of the use of these 8 parents don't have a lot of negative traits that will come along in crossing the traits that we identify of interest into local germplasm. So it expedites the whole breeding strategy. So the 8 parents were selected because they have some useful traits, and we know quite a bit about some of the parents in this particular population. I should mention that the International Rice Research Institute has developed several different magic populations. This one is an indica population that we'll be talking about today. There's also a japonica population, and there's an indica by japonica population with 16 parents. So the combinations can get quite amazing. So there are 3 crossing events that are used, and this has been published if you want to track how they did this population, but it's a huge undertaking, unbelievable undertaking, which requires a lot of tracking of individual lines. And the advantage of having these 3 crossing events is that you have not only increased recombination in the offspring, but you can uncover traits that were hidden by a genetic background, and you get tremendously improved mapping resolution and unique gene combinations. So if you think about it, what we've done when we get down here to the end and we're maintaining these progeny through single seed descent, 2,100 magic lines through single seed descent, you've scrambled the genomes of 8 parents. So you can imagine the power that that population has. So the plan that we had, and that Anna Bosa led this project, was to identify resistance to these African both bacterial blight pathogen and bacterial leaf streak pathogens. So we took the magic population, which was genotyped by sequencing. So all of the lines were genotyped by sequencing. We actually used a subset of that 2,100 and in fact demonstrated that you can get away with a subset because of the power of the population. We phenotyped them both in the greenhouses at CSU, which it was used to effectively screen initially the populations, and then we took the populations to fields in Mali and screened them again with both pathogens. And then because of the nature of the population, you can perform the mapping using both genome-wide association mapping, or GWAS, and this is a Manhattan plot showing the different chromosomes of rice with the traits showing up above the line. And then also you can use interval mapping, and that's powerful because we can confirm our QTL using both mapping strategies. So I'm just going to give a very short summary of a lot of work, and this is the map that we ended up with. So Anna was able to identify 14 different QTL, some of which were effective against a single group of isolates, so probably single gene related traits. But 11 of these were effective against both XOO and XOC, so bacterial blight and bacterial leaf streak. And we screened with over 20 isolates, mainly from Africa, but a few from Asia, because we were looking for African sources of resistance. And after using both interval and GWAS mapping, which interestingly, for the most part, identified the same QTL, which is great because we had validation of those QTL. You can see here, for example, this is one QTL that's of interest over here on chromosome 4. And you can see that it was effective against many strains, and that the stars indicate that it was that it was identified by both GWAS and interval mapping against that strain. So this is a very interesting QTL because it's effective against all of these strains from very different geographic areas and different pathotypic information. This QTL was effective against them. So the MAGIC was useful for identifying these novel broad spectrum sources of resistance. And given, as I said before, that these were elite parents, this can now be introduced into a breeding program in Africa and introduced into germplasm without a lot of genetic drag from other negative traits. Now, I won't talk about it today, but the second paper that was published this year, led by Alejandra Huerta and Emily DeLorean, we also used the MAGIC population to identify resistance sources against a virulence effector called TAL7B. It's a TAL effector identified from Xanthomonas oryzae. That is, it contributes to virulence in Xanthomonas oryzae, to bacterial blight, but is a minor effector. And we identified sources of resistance to TAL7B, and actually, because of the nature of the population and the lines that we identified with resistance, we were able to predict that the mechanism of how that TAL7B is effective against a plant, or the resistance is effective, is through loss of susceptibility. And that's in the paper by Huerta and Emily. So what we've been able to do using this wonderful MAGIC population and our collaborations around the world is identify some sources of resistance that target many strains of two very diverse pathogens, and then also identify sources of resistance that are effective against very specific virulence effectors, so likely single resistances that may offer increased durability in the field. So that's the first story that I'd like to tell you today. And the second story is that we've also been looking for a long time for quantitative traits that offer broad spectrum resistance to pathogens of rice. And as you know, many of you know, the pathways to plants' defense responses are diverse. With basal resistance or PAMP-triggered immunity, you will activate through surface recognition of conserved elements on the pathogens, or RG-mediated responses, which leading to effector-triggered immunity. But and both of these activate defense responses. But what's interesting is that the transcript profiles of PTI and ETI in different systems indicate to us that after resistance is activated, the defense response genes that are activated are very similar. They overlap. And so this is because many of these defense response genes are the enzymes or proteins that actively or collectively inhibit pathogen multiplication. Now, I liken this to a system where your effector-triggered immunity or your PTI are the generals that tell the soldiers what to do, and the defense response genes are the soldiers. And if you don't have good soldiers, if you don't activate good responses, it doesn't matter how much the general shouts if the responses downstream are not effective. So we've been looking for a long time at, if we enhance that basal response, can we increase the durability and the spectrum of resistance? And so the questions that we've been asking for the past many years are, what are the defense response genes that activate basal resistance, that function in basal resistance? Why are some genes more effective than others? And is genome-wide accumulation of these genes an effective way to go for resistance, improve resistance. So very quickly, over many years, we identified what are the genes that are controlled in shared defense responses. And these are genes that you would expect that underlie quantitative traits. And we got a consensus of these candidate defense response genes that were associated with QTL from multiple different populations, including different crops. And then over the years, we've asked individually if we mutate these genes, if we knock them down using silencing techniques, do we improve or do we lose resistance or in a quantitative way? And in fact, here's an example with a phenylalanine ammonium gene where we identified mutants in that gene. And the mutants in that gene had loss of broad spectrum resistance. So they were more susceptible to bacterial blight, more susceptible to rice blast, and more susceptible to sheath blight, which is a Rhizoctonia solani. So OSPAL4 was noted to be a broad spectrum disease resistance gene. And we did this with many different genes, asking if they played a role in broad spectrum resistance. But the big question really is if you accumulate these genes, can you enhance resistance? So using a different population, we identified lines that had QTL containing none of the defense response genes, up to 5 of these quantitative traits, which included different defense response genes. And we screened them in 5 locations across China and the Philippines, looking for the amount of disease over the accumulation of these genes. And what we found is the more regions with DR genes that we accumulated, the less blast disease we observed. So yes, accumulation of these genes does improve resistance, but is it durable or long lasting? So this actually, this is still ongoing at a limited level, but over the course of the experiments that we had done, we planted these lines out over many years. And in rice, you have 2 crops per year, so it's 22 cropping seasons, and tested the resistance to leaf and panicle blast, which is 2 forms of blast disease, in 2 different locations in China. And this was performed with collaborators at not only the International Rice Research Institute, but the Guangdong Academy of Agricultural Sciences. And here is our parent, our QTL donor in green. These are what this is, oops, sorry. The line that accumulated the 5 traits in red, and the susceptible recipient parent in gray. And you can see that over the course of the 22 seasons that we do have stable resistance in rice to that blast. So we then asked the question, why are some of these genes more effective than others? And the answer to this is that the difference lies in the promoter. And we've known this for many years, that activation of defense response genes occurs in a susceptible line, but it occurs earlier and faster and to higher levels frequently in the more resistant lines. So when we looked at the promoters of these genes, for example, this is an oxalate oxidase gene, which is a defense response gene in rice, we found that the promoters often either had insertions in our resistant donor or and that these insertions added cis elements. So what is the difference between an effective and an ineffective QTL? They both contain these defense response genes, the same genes. The genes aren't usually affected, but in the effective, they inhibit pathogens and they are broadly functional, usually against multiple pathogens. But the DR genes are regulated differently in the effective QTL. So there's either expression versus no expression or higher expression or earlier expression of those DR genes in the effective QTL. And we've shown that if we accumulate these effective QTL, that we can increase durable and broad spectrum resistance. So the question that we've been asking more recently is, does the cis element content of these defense response genes promoters in the effective QTL differ than in the less effective QTL? And our question really is, do the cis element motifs, which are present in the effective QTL, vary? And I see that I'm running short on time, so I'm going to sort of touch on this, and this work is published, so I'd be happy to have share the publication with you or answer any questions about it. But our hypothesis was that the shared cis regulatory architecture across broad spectrum defense response gene promoters is predictive of function in resistance. And this is chiefly the work of a PhD student, Brad Tonneson. So what Brad did is he assimilated data from 44 different expression experiments that were resistant versus mock inoculations, and also included some where we used where the published information used PAMP-triggered immunity. He normalized these and scaled them for a cross-platform analysis. He identified clusters of co-expressed genes, selected clusters enriched for defense response genes, and then identified motifs in those defense response genes, created a motif profile for each promoter in the defense response gene cluster, and then aligned the profiles to identify conserved arrangements of these cis modules, and then evaluated the module enrichment. And what he found, and I'll just give one example, is that, for example, in the phenylalanine ammonia lyase family members, these motifs, which are made up of groups of these elements, were enriched and present. So in this case, there's a motif 4 and a motif 2, which is underlined here, and it can be on either position. In the defense response genes that were active, the family members that were active in resistance and not present in the same architecture in the susceptible responses, and not present at all. So the CRIMs were in the promoters of the defense active QTL. So OSPAL4 was the active one, OSPAL2 is not active. And they are only present in the resistant alleles and not in the susceptible alleles of those promoters. And he did this for a number of different genes and showed that these associations are related to activation of the defense responses. So we've shown that early and strong activation of transcription of defense response genes contributes to broad spectrum resistance, and that the promoters of broad spectrum defense response genes are enriched in specific short sequence motifs or cis elements, and some of these are unique. And we've shown that groupings of these defense responsive cis elements, which comprise cis regulatory modules or clusters or groupings of these things, are enriched in the promoters of these broad spectrum defense response genes. And that alleles of the defense response genes demonstrated to be most effective in resistance contain these CRIMs. So now our question is, how do they enhance defense response gene activation? And we're using some silencing, some gene editing technologies to ask that question, but we're not quite there yet. But what I want to bring home today is why we care. And that is because if these cis regulatory modules can be used as markers and they're present in defense response genes around the genomes, they're shared around the genome, then we can use these as markers to enhance accumulation of the effective defense response genes and enhance broad spectrum disease resistance. So as Brad would say, is there a future breeding strategy for broad spectrum durable disease resistance that will be a genome-wide enrichment of these defense response genes? So finally, we've used MAGIC, these novel tools, to help us identify new sources of resistance. We're now trying to understand what those sources of resistance are and why they're effective against multiple diverse pathogens. And we've used a strategy of accumulating defense response genes to show that we can increase disease resistance and that promoters are important. And now we're beginning to understand that that can be a genome-wide strategy for breeding for disease resistance. So finally, I'd like to just touch on a few lessons learned over the years. I've been at this for over 30 years, so I have a few lessons, and this is just a few. And many of the lessons I've learned were the hard way. But it's important very early on, and this was a mistake I made in the early years, was not engaging with crop breeders. We assume we know what breeders want, but the issue is, do we really know what they want or how they will translate it? So I encourage you to engage with the breeders early and regularly. And also engage your students and postdocs with the entire team. It's a wonderful mentoring experience to bring students into large teams that span the globe. And I think they, not only do they make future colleagues by doing this, but also they learn a lot of lessons, good and bad, in the whole process. And you can also gauge from your students those that, some of them can just take the project and run with it, whereas others need a lot more coaching along the way. And then clearly, one needs to credit all team members, because all contributions, no matter how big or small, or what kind of contribution they are important and necessary to make these things translate to the field. So finally, with that, I'll thank my wonderful collaborators from around the world, chiefly in this project, the International Rice Research Institute, the International Center for Tropical Agriculture in Colombia, and the Institute for Research and Development in France, and also some universities around the world, and then the funding groups. And then this picture is an older picture of my group, but it brings many of the people that were involved in this particular set of projects to the forefront. So with that, I'll stop and take questions. And I'm sorry if I went a little over time here. So stop share.
No, you're fine, Jan.
Okay, thank you.
Just to remind people, if you want to ask questions, enter it into the Q&A box, and Jean and I will moderate those questions. So we have a couple questions that are very related to one another, and actually a question I had too that I wrote down. This is from Morgan Halani. Great talk, Jan. Are there any observable fitness or size trade-offs with lines with higher defense-related genes that are accumulated?
Yeah. You know, the yield trials have been ongoing on those lines for years, and we have not observed, again, there's a lot of work historically that shows that we're screening these under disease pressure. If we pull the disease pressure away, we may see some, but we have not seen compared, of course they yield better than the susceptible lines, but they also yield better than the resistant parent because they're more improved. They have a better genetic background than the improved parent. So I think that the answer is, so far it looks good, but we are screening under disease pressure.
Okay, so I have a follow-up question related to that. So in crops like corn and soybean and potato that I work on, it's yield, right? So no matter how much disease resistance we put in there, if there's any kind of yield gap, then growers just aren't interested in it. Is that different for rice, where you have a lot of small subsistence farmers that are just kind of growing rice for themselves or their family?
Well, historically, we have gone after yield, but in the parts of the world where rice is grown, there's such huge issues, and you're going to hear from Pam, flooding is an issue, and we had because we can't afford to use herbicides, which actually I take that back, because now we're going to direct seeded rice to save water. So they are having to use more herbicides, but these resource poor farmers really can't afford to spray a lot of chemicals on their crops. So we either have to put resistance in, or they don't get the yields, or they don't get sufficient production. And there are other, there's a combination of issues. It's not even just one disease at a time. You walk through a field and you'll see bunt, you'll see blast, you'll see. So it's critical to these people to get genetic resistance in. And also, when they do use antibiotics for these bacterial diseases, which is questionable that they're effective, they use them to an excess. And actually, there's a huge issue that's being brought up of antimicrobial resistance showing up because of the amounts of antibiotics that are being used in some parts of the world, surprisingly, on rice.
Interesting. I'm going to move on to our next question from Guido van den Akkerbecken, who asks, do you observe higher copy numbers of DR genes in your resistant rice lines?
In general, if they're there, they're all there. The genes are there, they're just not active. And we haven't, they are amplified. So for example, one group of defense response genes we work with has 12 copies. But the susceptible varieties have all 12 copies, but they're promoters in front, and not all 12 copies are active in resistance, which makes sorting all this out pretty complicated. And they contribute, there may be 5 of the 12 that contribute, and they contribute differently. They contribute quantitatively, but differently. Mm-hmm. And their expression levels are different. And so they're all there. It's just getting the good genes or the effective genes in place. And it's been challenging.
I was actually wondering, I'm going to sneak my question in, whether some of your cis elements, I don't know whether any of these genes are in different locations or if any of them are in tandem, and whether some of those elements could actually also be amplifying expression of the gene just upstream, functioning more like an enhancer.
That's a good question, and we know that it's more complex than our simple model puts it to be. And in fact, in the case where there's 12 genes, I think it's 10 of them are contiguous, and then 2 are off in another location. Yeah. So there could be some activation by the promoters in front of one of the genes, and we know that it's a little more complicated than we've simplified it to be, but we haven't gotten quite there yet. But that's actually an excellent question, and something we're ready to start to tackle in the near future, I hope.
Interesting.
So we have a question from Dimitri Lepin, and this is a question that's kind of plagued our community for a while, and this is kind of defining immunity versus resistance and basal resistance versus quantitative resistance. So he's asking what your definition of basal immunity is, or is there such a thing? Is there a basal resistance? Is it different from basal immunity?
So I always say that this is basal resistance. It's the resistance that's activated downstream of the signal that's sent by the PAMP-triggered immunity, and in my definition, and the ETI. And there's a lot of overlap in that. So that would be like the active oxygen species generation, oxalate oxidases, or enzymes that are degrading toxins, but also producing active oxygen species. So, and also, the genes that are involved in phytoalexin production are activated downstream. So that's kind of how I've categorized the basal resistance. And often the genes, those downstream genes, are encoded under QTL. We've demonstrated that several times for resistance. So they're differently regulated by the different mechanisms, but they're all part of that basal defense response.
Okay, I'm going to move on to Dave Berger's question, which is, do you get durable resistance to one type of pathogen, but increased susceptibility to another?
That's an excellent question. We could, but so far, in the cases that we've tested, we've seen a resistance to multiple pathogens and not enhanced susceptibility to others. But I have to say, we have not screened viruses on these lines. We've not screened insects on these lines. So there is a chance that we could, in fact, do that. But we have not observed it in the different pathogens that we've screened, which are blast, which is Magnaporthia oryzae, blight, bacterial blight, sheath blight, which is Rhizoctonia, and bacterial leaf streak, the three kind of major pathogens that we deal with. But we have not seen our fields go down with other new pathogens emerging in those fields. So I guess it could happen, but we have not observed it yet. Fortunately for plant pathologists, I don't think we've solved the problem completely. There'll be jobs for people forever, but I think hopefully we'll have a little bit more stability underneath those single resistance genes with these genes.
So Eileen, Jin asked the question, how stable are the defense-related genes in terms of maintaining their expression in following generations?
Well, so far, that's a very good question, and we haven't tracked it out past, probably we haven't tracked it out for all of the genes underneath in that line where we accumulated the 5 different QTL. We haven't tracked it out further than probably the 8th or so generation. So if you have a lot of pressure on the evolution in the field, you could select for evolution towards susceptibility, but I think it's a little slower than it would be for a single gene because the pressure is not so great on these because there's a contribution of multiple genes to it. But again, we haven't tested it out that far. It's going to be maybe another 20 years before we have that answer.
A bit longer for a graduate project.
Yeah.
Okay. Michael Lassner asks, do you have any hypotheses about the origins of the activating cis elements?
Well, we have some ideas. Some of them are novel, but most of them are ones that people have already identified as defense response active elements. And why why these modules have formed and why they're inherited, my hypothesis would be that the module forms and it's a positive factor for the host to have these because these genes help protect the host, and so they've increased or improved in the population. Actually, I also think that breeders have carried them forward without realizing what they were carrying forward, because they improved the disease resistance in their host in the plants. But there's an interesting story that we're following up on, and I'd be happy to talk to you sometime when we have more time, where the Xanthomonas pathogens use these TAL effectors to drive resistance, and we're seeing some association of some of the effector binding elements with some of our crims. And we don't know what that association means yet. It may mean nothing, but it's interesting enough for us. I have a master's student who's sort of playing with some of the bioinformatics in that right now, trying to understand if it's meaningful or not. But if it is true, it could be that there's a relationship between the activation and the suppression of defense response genes, that the pathogen tries to suppress them with these effector binding elements being present, but the plant evolves a way around it by putting other elements in contiguous to those or around those. Purely hypothetical and no foundational information, but kind of intriguing.
So we have a few questions related to what you think your next steps are with using the CRM data and promoters. Could you potentially do promoter swapping? You know, would these perhaps prefer or confer durable resistance? Are you going to go to the chromatin level? What are you thinking the next steps would be?
So we're doing some editing experiments right now to ask how the structure of the crim, certain crims are important. We're also working with some breeders at CIOT. We've just started a project, hopefully, where we'll see if we can use that information to develop markers to do genome-wide breeding, because we know that the crims are in front of different defense response genes, not just the same gene family, but around the genome. So if we could design markers, could we use these to effectively improve for the good, the effective QTL or the effective defense response genes under those QTL? So that's really my passion, is seeing if we can take this to that next level. And but we're also still trying to understand how they're acting and why they're effective, and why they're inherited as they are. But my passion is really to see, can we take this all the way to improving breeding strategies? Because if you think about it, it's not just disease resistance that this is probably true for. In fact, that's one of our hypotheses, is that it's also true for drought stress or temperature, heat stress, because these are stress responsive genes and they're activated by across the genome. They're simultaneously activated or by different stresses. So if we're correct, this could be a new breeding strategy for improving for stress tolerance.
Yeah, I think our next question, things got moved around a little bit, sorry. Maria Florencia Ercoli asks, have you done promoter swapping considering the data from CRM?
Mm-hmm. So we have not. We're doing it by editing, but it would be nice to do that. Rice is not as easy to do editing in as I would like it to be, but we're doing some attempts at taking the promoter, those insert regions from the good effective genes and putting them into the less effective genes and seeing. And we're just getting lines now, so we should have some of the answers to those questions. But like Jean was saying, it might be more complex. They may be driving things, there may be things upstream that are important that we don't know about, but at least we're starting with those.
All right. So a question from Anonymous. Have you seen cases in which expression of your defense-related genes is enhanced, but initial perception of the pathogen is the bottleneck, missing the general?
Mm-hmm. I don't know that we would have recognized it if it occurred. Because a lot of what we're looking at are PAMP-triggered immune responses. And I'm not sure that we would have recognized it if that upstream signal was missing, because there may be multiple signals feeding into that. So like if you have multiple PAMPs, you're going to trigger, from different directions, the pathway. So we maybe would not have recognized that, but I'd be happy if you have an idea for an experiment to think it through.
So I have a question that takes you off in a different direction. We've been wondering how in this year of pandemic, could you just comment on how you're navigating lack of travel for these huge international projects?
I haven't been on an airplane since March, and for someone who travels over 130,000 miles a year, that's quite a quite a withdrawal. And what has happened is that it's really taken Zoom to a new level, or Teams and whatever. And what I find is that while we're maybe in connection more frequently, it's not as meaningful a connection, because it's one thing to look at rice on a screen. Behind me you can see rice. It's another thing to walk through a rice paddy and see the different pathogens and pests that are there. So it's been trying and difficult, but I hope it's I think of my students and my postdocs. I hope it's given them the opportunity to kind of sit back and evaluate how to more efficiently and effectively work internationally. But there's nothing like a face-to-face, I have to tell you.
All right, we've got time for maybe one more question here from Nick Bird. How many loci can be tracked in a breeding program effectively, cost effectively, while still delivering an appealing overall package?
That's an excellent question, and I am not a breeder, so I'm hoping that maybe somebody in the audience can chime in and answer that question. I honestly don't know.
So we have some other terrific questions. We're going to stop there, I think, right, Dennis?
Sure,
and
yeah.
I think that they'll be able to, our support here should be able to save the questions you didn't get a chance to answer, and maybe we'll be able to address those later. Thanks so much, Jan. That was terrific talk, really exciting and interesting. I think should also be great for students who are just entering this whole field. So I'm going to turn it over to Mary Beth now.
Thanks, Jan, Dennis, and Jean. That was wonderful.
Thank you.
Well, it's a real pleasure for me to introduce Brian Staskowitz, my mentor and my inspiration. Brian Staskowitz is a professor at the Department of Plant and Microbial Biology at the University of California in Berkeley. Brian is widely considered to be one of the most influential scientists whose research and mentoring has really left marks in the field of MPMI. His seminal research accomplishments include the cloning of the first bacterial effector genes and plant disease resistance genes when folks thought you couldn't clone those. These are landmark achievements in the field of MPMI, as well as animal immunology, providing the first experimental validation of the existence of pathogen and plant genes to support the ongoing gene for gene hypothesis, which was the central dogma in plant pathology established by Harold Ford. Throughout Brian's career, he has focused his work to engineer durable resistance in several crop species, including tomato, pepper, cassava. He's dabbling also in cacao. He has pioneered several genetic approaches to exploit natural variants in disease resistance in host populations using map-based cloning strategies, as well as mutagenesis. He's shown that you can have gene transfer experiments of resistance genes that can be transferred across genera and retain their function and specificity, which is really opening the door to plant engineering approaches. Notably, Brian has always been at the forefront of technology development. His interest in that and driving it has shown to be really critical for scientific discoveries and their translation. He's currently the scientific director, co-director at the Innovative Genome Institute in Berkeley, and he's leveraging the use of CRISPR gene editing technology to optimize traits in a wide range of agriculturally important crops. The title of Brian's talk today is Engineering Durable Resistance in Crops. Welcome, Brian. We look forward to hearing your updates.
Thank you, Mary Beth. So I can share the screen now?
Yes.
Everybody can see that?
Yes.
And we'll go to presentation mode. Okay, great. Well, this will be kind of a continuation of some of the themes that Jan just presented in her talk, but we're going to talk about our strategies for creating durable and sustainable disease resistance. And the system that we've worked on for many years now, is the bacterial spot disease of pepper and tomato. And as many of you probably know, there are many species of Xanthomonas that can cause disease on pepper and also on tomato, including U. vesicatoria, Gardneri, performs, and others. So that's the disease. Okay. So here's our approach. This is the outline for my talk today. So I'm just going to talk about some published information, how we've used a pattern recognition receptor, the EFR, to recognize the EF2 PAMP from Xanthomonas species in tomato, how we are currently using CRISPR-Cas9 genome editing to construct precise mutant alleles that confer resistance. I'm going to talk about a recessive gene called BS5 and a gene called DMR6 for downy mildew resistance 6 that was originally identified in Arabidopsis. I'm going to summarize some of our work on where we can actually identify the core effectors in Xanthomonas to guide the use of cognate resistance genes, where we've used the BS2 resistance genes, and how we can use these effectors to discover new R genes, in this case, the ROC1 gene in other solanaceous species. And we really feel that a combination of pattern recognition receptors and multiple R genes that recognize multiple type 3 effectors, along with recessive resistances that lower bacterial populations to reduce the chance pathogen mutants will be selected in the field, will provide durable resistance. And at the end, I'm going to present unpublished data that will, I think, gets published this Friday in Science, where we have actually solved the cryo-EM structure of the ROC-ZOP-Q resistosome, which then gives us many insights into the possibility of creating novel synthetic resistance genes. So here we go. Along with Cyril Zipfel and other collaborators, we published several years ago that you could put the EFR gene in the tomato by transgenic, and you're able to get resistance not only to Xanthomonas, but also to Ralstonia. So basically, the gene was just done by agro, and it really works quite efficiently against Ralstonia in the field. So that's one approach. Another approach is really using gene editing to actually identify new resistances. So our strategy here is to identify previously characterized mutants in other plants that provide bacterial resistance. For example, the DMR6 gene in Arabidopsis thaliana and the BS5 genes in tomato. And then to use genomics and to identify orthologs in tomato and create CRISPR-induced mutations in those respective genes. So the first gene was originally identified by Van Dam in 2008 in Guido van den Akken's lab in Utrecht, where they first identified in Arabidopsis thaliana an upregulated gene called ATDMR6. They showed that it accumulates more salicylic acid than the wild type plants after infection. And at least in the case of Arabidopsis, there was no clear detrimental effect in growth and development. So, knowing the sequence of that gene and having the genome of tomato available, one is able to actually search the genome of many crops, and you can find the orthologs. And you can see in this particular situation right here, this is the DMR6 clade. And if we come over to here, we'll see that there are 2 copies of DMR6 in tomato. We call them DMR6-1 and DMR6-2. So I'm not going to go over the basics of CRISPR. Many people know this. But basically the idea is that it's relatively straightforward, very straightforward in tomato to make mutations in genes. And all you have to do is identify target sites using various algorithms, design guides, do agrobacterium transformation containing Cas9, and then eventually select the mutations. And so we're able to get mutations at various locations, either exon 1 or exon 3. And I wouldn't be telling you this otherwise, but obviously it works. Now, one thing that's interesting is that I said there were 2 genes in tomato for DMR6. So DMR6-1 seems to be expressed in the roots and also in the leaves, where DMR6-2 seems to be expressed mainly in the fruit. And that's kind of borne out. And when we actually look at make mutations in either DMR6-1 or 2, you can see that DMR6-1 lowers the bacterial growth here when we're looking at the log growth right here, but DMR6-2 did nothing on that particular case. Also, you can see that when you knock out that particular gene, you increase the total salicylate that's actually in that plant, where DMR6-2 did not do that. Now, Daniela Tomazella, who was a postdoc in the lab at the time, purified this particular protein, and there have been some controversy of the biochemical role of this particular gene product. But we are conclusively showing right now that this will soon be published, that basically DMR6 encodes for a salicylate hydroxylase. In this particular case, you can see it up here. And that I'm not going to go through the various controls here, but you can trust me that all the HPLC patterns give rise to the fact that they do both. So DMR6-1 and DMR6-2 encode salicylate hydroxylates, but the DMR6-2 is only expressed in fruit tissue and flowers and not in the rest of the plant. So what you have here is an interesting situation where, and I should also mention that these genes originally shown by Guido's group, is that these genes are only expressed, the DMR6-1 genes are only expressed around invading pathogens. So plants have evolved a way to actually lower their salicylate around infection sites. You would think just the opposite, but they have to have some homeostasis of salicylate. So what we show then is that these mutants show broad spectrum resistance, not only to Pseudomonas syringae, Xanthomonas gardneri, perforans, Phytophthora, and also powdery mildew. Here you can see that the wild type is just covered with powdery mildew, and here the DMR6-1 is basically clean in this case. So the next question you're going to ask, what happens when you put these in the field? So our original field trials have shown that there is definitely some growth reduction, and there's some penalty for producing salicylate in these particular plants right now. So what we're currently doing is that we're actually going and making new sets of mutations in the promoter of DMR6 to basically try to find a rheostat of activity from totally off to overexpressed, and see if we can find a sweet spot where we can get enough salicylate production where we don't actually see any negative growth phenotypes. So DMR6 is a CRISPR approach to try to get broad spectrum resistance. Now, another approach is to actually use a gene that's been identified in pepper. So work by Bob Stahl and Jeff Jones for many years at the University of Florida had shown that there are 2 pepper recessive resistance genes, one called BS6 and one BS5. Now, what's interesting about these particular genes is that they're not the typical, when you inoculate Xanthomonas into these leaves, usually you would see a hypersensitive reaction. But in the case of BS5, which is this ECW, early Cal Wonder 50R here, you can see that what you do is you just reduce the lesions. You don't really get that. So we don't know what the mechanism of resistance is, but we thought we would ask the question, can this gene be cloned in pepper? And since pepper is a solanaceous plant, are there corresponding orthologs in tomato that we could actually gene edit to give rise to this particular phenotype? So work done by Jorge Kiss in the World Vegetable Center in Hungary. Basically, this is their work where they actually did a map-based cloning. And again, I'm not going to go over the actual. So what you basically do here is you have a resistant plant, a susceptible plant, and then you identify markers, and then you find recombinants, more recombinants, till you narrow down the particular gene in this case. To make a long story short, the gene was identified. Now, this is a very strange gene. If you look at it, you'll see that basically it's highly similar to a neighboring gene. I'll show you that in a moment. And it only encodes 92 amino acids. It has this 5 amino acid repeat, and it's very cysteine rich in the C terminus of this particular plant. And this family of genes has been shown in Arabidopsis. They seem to be important for development and response to stress. Now, what's interesting is the nature of the mutation in the BS5 allele is a 2 amino acid in-frame deletion right at the C terminus of the gene in this particular case. So if we look at the gene structure on chromosome 3 in the Pepper genome, what you have is the BS5 gene. And then we have what's called the BS5-like gene. And you'll see that it's almost identical, but not. There's some differences. And the difference between the wild-type BS5 gene and the mutant recessive BS5 gene right here is this 2 amino acid deletion, in-frame deletion. So you still get a protein made, presumably. Now, the BS5-like gene in this particular case is missing one of these repeats and is also has the 2 leucines. So we don't know exactly what role these particular genes play. So we're interested in determining that, but pepper is a very difficult plant to transform and to do gene editing in. So we then said, okay, let's go to tomato. So what we did is to find the orthologs in tomato, and you can find the same gene arrangement in tomato here. So this is the BS5 ortholog here. This is the BS5-like gene, and these particular genes are flanked by the similar types of genes that are found in pepper. So there's synteny between pepper and tomato in this part of the genome of both pepper and tomato. So typically, the same strategy here is that ideally what one would like to do is to be able to do allele replacement, but that's pretty difficult in tomato. So we asked the question is, can we make mutations in these genes and do we see a phenotype? So again, knowing the sequence of the gene. So it's essential for gene editing, you have to have a genome sequence. So you need to have that so you can actually identify targets, but also more importantly, off targets that you don't want to actually make mutations in. So basically, you introduce the guides and the CRISPR system into the plant cell, you regenerate plants in this particular case. And we found a couple of lines, line 1 and line 2 in this particular case, that actually had, in-frame, they basically had mutations that caused premature stop codons, and so we got truncated proteins in this particular case. And to our surprise, it turned out that you don't have to have that particular 2 amino acid in-frame deletion, that a mutation in that BS5 gene seems to give rise to the same type of phenotype here, where you get about 1 to 1.5 log reduction in growth. So we feel that this could be a very effective gene. And we're excited about this particular gene because the BS5 gene in pepper has been in production in Florida for over 20, 25 years, and it's never broken down. So it's a recessive resistance. So we're hoping that as we continue field trials with our collaborators at the University of Florida, that this gene will become useful. And if you think about it, taking these genes and combining them with other genes could be very advantageous. So here's our other strategy, which is effector-targeted breeding for bacterial spot disease resistance. So the knowledge of effector content in natural field strains of Xanthomonas informs the potential stability of disease control in which our gene should be deployed to achieve, hopefully, if it's not durable, but longer lasting resistance. As Jan so nicely showed, is that anytime you put a selection pressure on a single gene, it usually breaks down quite rapidly. As you know, the pathogens find ways to which to either knock out the gene or modulate it or to do something to actually get around the recognition of that particular effector. So a graduate student, Alison Schwartz, in my lab did a very nice study where we identified a whole bunch of strains. And these days, I don't know if you remember when the first Xanthomonas sequence came out of Ulla Bonus's lab, it cost about $1 million. Now you can do it for $6, you can, or less now, you can sequence, you can just really sequence a lot of strains. And so we have a pipeline where we can actually multiplex sequence, barcode these, sequence these particular genomes, identify the effector contents, and identify which effectors are in which strains in which fields. And you can monitor this in the field over time to actually inform what types of resistances would be important to actually deploy in the field to control those types of resistances. So I'm not going to go over this particular one, but we identified a whole series of genes, and I'm going to talk about two today, just to keep it straightforward, is that I'm going to talk about AVR-BS2, which is a gene that is recognized by BS2, the pepper gene. This is a gene that was originally identified in Capsicum chuckoense, in a wild species of pepper. It's called bacterial spot number 2, and the effector is called AVR-BS2. And just for historical purposes, AVR-BS2 was the very first effector that was shown that it actually had a virulence effect. So for a lot of the students in the audience, when we first cloned these genes, people would say, why would pathogens have a gene to trigger host defense? Well, as we well know right now, effectors really either modulate or suppress the immune system in plants. So AVR-BS2, when you made a precise gene knockout of that particular gene in Xanthomonas, you got a lower growth. Not true of many of effectors. When you actually knock those effectors out, you don't see a growth. So it's probably the culmination of a whole series of 20 or 30 effectors that give rise to the ability of a pathogen to cause disease on any particular plant. Now, another very interesting gene that's in all strains of Xanthomonas is ZOPQ. And it's actually in most pathovars of Xanthomonas also. So we're going to work with AVR-BS2 and ZOPQ. So this, again, was one of the examples where when we first proposed to do these experiments, people didn't think that it was going to be successful, in that there's no way you're going to put a pepper gene in a tomato genome and actually get disease resistance in this particular case. But since it's effective, we're able to do that, and it showed it very nice. So actually, many field trials over many years now have been done with our collaborators, Bob Stahl and Jeff Jones, and down in Florida. And basically 92% of Florida and 40% of the US tomatoes growing area is affected by bacterial spot. Chemical control is inadequate. The way they try to control this disease is by copper sprays. And as Jan mentioned in her talk, as you can imagine, when you put a selection pressure on for copper, you're going to get copper resistance. But it's even more crazy than that. It turns out that copper resistance is on a self-transmissible plasmid within Xanthomonas. So using chemical control, what you're doing is you're selecting for a plasmid to actually expand throughout the population of Xanthomonas. And even more amazing is on those plasmids are transposable elements, which we have shown that can hop in and to inactivate an effector gene. So by spraying chemicals, you're actually mutagenizing the population of the bacteria, not only for copper resistance, but for actually overcome disease resistance. Pretty interesting situation. So we showed Thomas Tai, who was a graduate student in my lab many years ago, showed the expression of this BS2 gene confers resistance to bacterial spot disease and pepper. So BS2 is a part of what's called an NLR gene for nucleotide binding leucine-rich repeat, and it has the flavor that it has a coiled cola domain. We now know that BS2 is not just a single gene, it works in a network, that BS2 is a central gene, and it works with the helper genes called the NRCs. I'm not going to go over that. That's a lot of work that's been done in Sophie and Kamoon's lab. Tomato and Nicotiana benthammiana transformants demonstrated an HR to this, Xanthomonas containing AVR-BS2, and bacterial growth suppressed in T2 tomatoes expressing BS2. And the effector is really universal in Xanthomonas perforans. So once again, BS2 in the field, a wild type without it, you can see there's an incredible difference. You can see all the fruit here. The fruit here is basically suppressed. And this was published by Diana Horvath in combination with Jay Scott. And we showed that it confers effective level of control of bacterial spot resistance. And what's more amazing is that with the BS2 gene, we were actually double the yield of marketable fruit in those particular plants. Now you have to ask the question, why was this never adopted by the Florida tomato growers? Well, most of the tomatoes that are grown in Florida either go to Walmart or to McDonald's. And the anti-GMO contingent really threatened to basically boycott McDonald's and Walmart if they were started selling these. So this material never got off the field. So this is a problem. I would beg to say that many diseases around the world, we could control diseases if we were able to use GMO techniques. More importantly, this is for late blight of potato by Jonathan Jones and Mark Ghislaine in Africa. There's a whole series of really great experiments. So it's a challenge out there. I think one day this will probably be overcome. And I think you're going to hear that Brian Stephenson at the end of the last talk's got a beautiful story by making transgenic durable wheat resistance to stem rust. So here's our story with ROC1. So it was shown here by Ulla Bonus's lab that non-host resistance induced by Xanthomonas effector ZOPQ is widespread within the genus Nicotiana and functionally depends on EDS. So what Ulla's group did in those days was to use virus-induced gene silencing and to show that when you silence EDS1, you now could convert a wild-type Xanthomonas strain that had ZOPQ in it to cause disease on Nicotiana benthamiana. Okay. So first experiment, this is an experiment that Alex Schultink, when he was a postdoc in my lab, was to delete ZOPQ from the Xanthomonas. And when you knock ZOPQ out, and similar work can be done by Alan Colmer at Cornell University, where there's a HOPQ, which is an ortholog in Pseudomonas syringae DC3000. So when you knock that out, you can convert Pseudomonas syringae DC3000 from a non-host to basically causing disease on benthamiana. The same thing can happen in Xanthomonas. So here it is again. So we thought instead of using VIGs, since we're in the business of making many CRISPR-mediated mutations in various crop species, we decided to make an EDS mutation. So this is important for many reasons. You'll see that this was key, actually, as I'll show you, that we wouldn't have been able to identify the resistosome by cryo-EM if we didn't have the EDS1 gene. I'll explain that more later. But we did here in this particular case show that EDS1 really controlled this resistance. So the mutation in the EDS1 allows Xanthomonas species to cause disease, as you could expect. Don't need to go over this in particular phase, but the idea is that, and we know that EDS1 is responsible for a class of NLR genes that have the toll interleukin-1 receptor. So how do we get ROC1? ROC1 had been hypothesized to be a non-host. We can discuss that in the questions and answer. What's non-host? It's probably a bad term, but anyway, people have used it. Non-host NLR immune receptor that recognize conserved effectors, HOPQ and ZOPQ in tomato. So when you actually use Agrobacterium tumefaciens that translantly express ZOPQ into Nicotiana benthamiana, oftentimes you don't get a very strong hypersensitive reaction. So Alex basically used this surrogate assay where he also put in TMV clone expressing GFP. So it's a surrogate. So when you see GFP, there's no recognition, and no GFP, there is recognition in this particular case. So again, here's our various classes of NLR genes. And again, this is expanded too, but we have the TNLs and basically the helpers, and we have the CCNLRs in this particular case. So knowing that EDS was controlling resistance, but at the time when we did this, there really wasn't a well-annotated NLR genome, if you want, or clones of all the NLR genes in Nicotiana benthamiana. So we collaborated with Jonathan Jones's lab to actually use what's called RenSeq, which is basically a bait capture of, you basically make RNA probes and you pull out all the NLR genes out of the genome, and then you do next generation sequencing and assemble these and you get a whole series of genes. And so then we can make the entire system show us that we have all the NLR genes. And what you have here in yellow are the TNL genes. So we thought that these were all potential candidates here to be the ROC1 gene. And Alex was a very unique postdoc. He said, well, I'm just going to go knock out every single one of these genes and find out which one is ROC1. And I said, yeah, that sounds good. That's going to be a lot of work. And it was a lot of work, but Alex did it. And what he did is that he found a particular clone that when compromised, we predicted that it was a TNL gene. There was a limited number of predicted TNL genes in benthy and allowed us to design, in this case, we use virus-induced gene silencing. So Nicotiana benthamiana using tobacco rattle virus, very efficient gene silencing. We could silence all 30 plus predicted candidate genes and find out which particular gene did not recognize ZOPQ. So tobacco rattle virus constructs containing the sequences, and he also kind of multiplexed this where he simultaneously targeted 3 to 4 TNL genes and then could deconvolute that and go back and find the one. There's a single gene that was eventually identified that was called ROC1, resistance to ZOPQ and HOPQ. These is all published right now. So this just basically is a slide. We know that ROC1 associates with ZOPQ and co-IP experiments. And specifically, ZOPQ interacts with the LR region of ROC1, shown by this particular co-immunoprecipitation experiment. I'm not going to go through the details of this, but again, this is published. You can see that. So it turns out that there's 123 pathivars of Xanthomonas campestris in various subspecies, and many of them contain ZOPQ orthologs. And Alex went and showed that many of these are actually recognized by ROC1. So potentially ROC1 can be a resistance genes in many other species. So one particular plant that I'd be interested in is to put it in walnut. One of the major limiting factors to walnut production is Xanthomonas juglandis. And so we haven't put ROC1 into walnut yet, but I think that's certainly a possibility. Expression of ROC1 in transgenic tomatoes conferred resistance to Xanthomonas. Also, it conferred resistance to DC3000. So HOPQ in DC3000 is recognized and you get very good resistance. And these have been tested in the field now and look very stable. Also, Ralstonia has an effector called RIP-B, which is an orthologous gene to ZOPQ. And ROC1 also is recognized as RIP-B in Ralstonia. So Ralstonia is a kind of an emerging pathogen in the tomato fields. And so this could be a very effective resistance against Ralstonia. And Alex has shown very nicely with various bacterial growth curves that you can suppress Ralstonia when you have ROC1 transgenic tomato plants. And you can see right here that the wild type is wilted and totally wiped out, where the ROC1 is totally unaffected. So it's definitely a very strong resistance. All right, the last part. So knowing that the ROC1 gene specifically co-immunoprecipitated with the leucine-rich repeat region of the ROC1, we were interested to know whether we could identify a protein complex in Nicotiana benthamiana. Now, what was essential, I told you previously, if you co-express ROC1 with ZOPQ and Nicotiana benthamiana, you get a hypersensitive reaction. So it's not a great protein expression system. But we knew that in an EDS1 background, we still could get co-immunoprecipitation of ROC1 and ZOPQ. And so we hypothesized that the oligomerization of the leucine-rich repeat region would actually occur in an EDS1 background. So inoculating the EDS1 mutant, we could then do, in this particular case, believe it or not, they did about 300 plants to get enough protein to actually get enough protein where we could solve the structure. So here's the structure. This will be published in Friday in Science. And I'll just give you another there's another TNL gene called RPP1 from Arabidopsis. And this was done by Chi-Chi Chai and Paul Schulze-Liefert and Jane Parker. That will also be co-published in the same issue. So be looking out for Science on this Friday. But what we showed very interestingly is that you form this tetrameric, beautiful structure in this particular case, where here you can see the various domains. This is the toll interleukin 1 or the TIR domain, nucleotide binding, Wien helix, LRR. And then there's a very interesting thing that we discovered, that there is a interesting post-leucine rich repeat gene, which we're going to call C, And this, I hate these acronyms, but we had to come up with something. It turns out there's a jelly roll fold, or as you'll see, there's an immunoglobulin fold of this particular domain. So you get this beautiful structure. You get binding at the leucine-rich repeat. This triggers, here's probably a good review right here, is you get recognition of ZOP-Q. You get this oligomerization of ROC1 containing the tier domain here. And what the tier domain does, what the beauty of this is that the tier domain basically is a dimer of dimers in this particular case, and it moves this BB loop down, which now allows for NAD to actually come in. It had been shown previously last year by Boston Kobe's lab, Jeff Dangle's lab, that this is particularly interesting because there's an NADase activity. And so the only way you can get NADase activity is you get this specific oligomerization of this tetramer of the tier domains, which allows the NAD to come in and bind in this particular site. And you get this immune response as a downstream. So this is a very beautiful, there are many unanswered questions exactly what happens after all these particular cases. We could talk about that in the discussion. And Raul Martin, who's the graduate student that did the majority of this work, is a biophysics student at UC Berkeley and Ava Nogales' lab, and did a great collaboration with Tian in my lab, who's a postdoc. And I'll just show you this beautiful movie that he actually showed, that he made. So you can see the tier domain, the nucleotide binding, and this particular, it's all color coded. So the leucine-rich repeat is in purple, ZOP-Q is in this salmon color, in this particular case. And so recognition of ZOP-Q is by the leucine-rich repeat and causes the oligomerization of ROC1. Oops. So as I told you that we getting close to time here now, the carboxy terminal portion of the leucine-rich repeat has this post-leucine-rich domain right here. So it turns out that we never could have predicted this unless we had the structure of this particular region of the domain of the leucine-rich repeat. So this PL domain has this conserved, and as you'll see, this variable domain. And what's amazing about this is that ZOP-Q is predicted to be a nucleoside hydrolase. No one has been able to actually show the substrate for that, but it's predicted by that there's an active site in there. But it turns out that ZOP-Q not only triggers the oligomerization of ROC1, but it also goes in and binds into the active catalytic site of this nucleoside hydrolase to actually inhibit virulence activity. That's been shown in HOP-Q by Geeta Coker at UC Davis. And if you start to look at the structures of this, you'll see differences from the variable domain, and you look at antibodies. And in this last slide right here, you can see that what you have here is essentially you've got conserved domains, and then you have this loop that goes into the ZOP-Q. So we're thinking right now that, and it turns out that other TNLs have these particular post-leucine-rich repeat domains also. You'll see there's a recent paper published in MPMI on this. That's very nice work. And you'll see that the RPP1 has one, and I predict all TNLs are going to have this particular structure. So what people have done in the immunology field is that they're able to use synthetic biology to essentially use phage display to actually run synthetic DNA libraries to change binding specificity of these particular domains. So the idea is to maintain the conserved domains, but then change out the variable domains, and then basically do a phage display and try to find new effectors that we can recognize. This would be the holy grail for many of us, is that we really would like to design synthetic resistance genes against any effector. That could be quite exciting. So this is all very new and exciting going forward. So just to summarize, what I've shown you today is that using pattern recognition receptors, making mutations in other genes that control resistance like DMR6, the BS5 gene, using dominant disease resistance genes like BS2 and ROC1. I really feel that if we can actually deploy multiple levels of immune receptors, both PRRs and other recessive genes, that maybe we'll have a better chance. But I'm also not naive to realize that pathogens find ways to get around host resistance pretty easily. So many people to acknowledge here, many people in my laboratory that are all in blue here. A shout goes out to Allison, and this Doug Dahlbeck, who's been involved with many of the project, Arturo with the BS5, Raul Martin for solving the structure of the ROC1 resistosome in Ava Nogales' lab. Our collaborators along for Jeff Jones, Bob Stahl, Jay Scott, Gary Vlad, and Sam Hutton in Florida. Our collaborators in Hungary, Jorge Kiss and Zoltan Szabo. Alex now is an entrepreneurial fellow where he started a company called Fortifight. Two Blades, Diana Horvath and Roger Friedman, who have really supported this research over the years, also a USDA and also the National Science Foundation. So I'm happy to stop there and answer any questions.
All right, well, thank you, Brian. As a biochemist, it's so exciting to see that new structure. So can't wait to read science on Friday. Looks like we've got several questions and different topics, so we'll try to organize them based on topic. If we go to the beginning of your talk, when you were talking about transferring genes from one plant species to another, the question from Dave Berger is really thinking about EFR. So do all tomatoes lack EFR orthologs, or really what's going on there? Why does tomato don't have EFR?
Yeah, that's a good question. It turns out that this tomato does not have EFR orthologs, so they're only in the Brassicaceae, like Arabidopsis. So you can find them in other Brassicaceae, but you can't find them in the Solanaceae. So basically, taking that from Arabidopsis into solanaceous plants seems to have worked very nicely.
So Roger Wise asks what complexes interact with the 2 amino acid deletion and their effect on resistance. So this is going back to the BS5.
Right. So that's a great question. So the BS5 gene is a very strange gene. As you saw, there's only a few, less than 100 amino acids, the 2 leucine. So we had some suspicion that it might have to do regulating type 3 delivery into plant cells. So we have some preliminary data now that those mutants using the adenylate cyclase reporter for delivery of type 3 is greatly reduced in the BS5 mutant. So we're very much interested to determine if that somehow is facilitating, that protein's involved with the delivery of type 3 into plant cells. We don't have any definitive answer, but that's something that we're following up with now.
And staying with that topic, Brian, is BS5 present in other plants? What's its range? Wheat, rice, or even outside of angiosperms?
So BS5, we know that it's in citrus, and we're trying to edit that gene in citrus now for citrus canker. So we don't have any results yet, but it's definitely, they're potential orthologs in other plants, but we don't know whether they're truly functional orthologs.
So keeping with this theme, I'm going to just jump down to Hailing Jin's question. Exciting talk. She says, do you know the mode of action of BS5? Is it secreted? And do you think it behaves like an antimicrobial peptide?
You know, it's predicted to be an integral membrane protein as a dimer, but you know, that's only hand waving. We don't have any evidence for that. So we really don't know what BS5 does. So that's the short, sad story about that. So sorry.
We have a detailed question from Arsh asking, it looks like when you were looking at the BS5 gene, your target guide RNAs were selected for exon 2 and 3. Why not from exon 1? Can you give us some insight of your strategy for doing this?
Oh, those are the ones that just guides that we tested first that seemed to work in vitro. So we just went with that one. That's not to say that exon 1 wouldn't work also. So I think exon 1 would work just as fine.
I have a question for you.
Go ahead.
I'll jump in here. I was really interested in the DMR6 story where you were looking at the expression patterns were really different between, say, the roots and fruit. And so you showed us really nice resistance that came out of your studies. But What was going on with the fruit and the importance of SA and development and senescence?
Yeah, that's a good point. We haven't really followed that up. So that's really not our data. That's what's publicly available data for transcriptomes that are available in tomatoes. So we haven't really followed that up. So yeah, I don't know what the significance of that is.
But did you get good fruit on your transgenics?
Say again?
What was the fruit quality on the
Oh, the
plants
fruit,
that you made?
yeah, the fruit looks fine, actually.
Ah, interesting. Okay. We have a question from Dimitri Lapin about the BS2 and DMR6 use in tomato. Do you notice if certain genotypes are not suitable for this introgressions?
Again, Dimitri, we don't know. We've only done it in a few tomato genotypes, but every one that we've introduced BS2 in. So we work with kind of an advanced breeding line that Sam Hutton has at the University of Florida, and it seems to work in that one. So we haven't extensively tested it in many different lines, so we don't know the answer to that, but definitely BS2 should work. And I think DMR6 should work also, but I think the warning with DMR6 is that the high levels of salicylate seem to give some stunted growth. And so I think we have to somehow tamper down that particular mutation and maybe make some promoter mutations in that DMR6 gene.
So we have another question here. Do you think that the transferring of our genes from one family to another would work if the genes are somehow recognized by other species?
Well, you know, people always make fun of me. I came up with this crazy terminology called RTF, restricted taxonomic functionality. I did that as a tongue-in-cheek kind of system. So for instance, we put BS2 into Arabidopsis and it doesn't work there. We have the capability of assaying that that's the case. And we think we know why now, because remember, BS2 is part of a complex of genes. Not only do you need the sensor of BS2, but you need the helpers, the NRC 2, 3, 4 downstream genes. So I think we have a lot of lessons learned if we're going to do wide-ranging transfer of genes. Maybe in the case of ROC1, which is a direct binder of an effector, it may not need we know that it does need a helper. It needs NRG, which is a CC NLR gene. So the TNL story is, you know, helper and sensors. This is an emerging story. And also, if people have been reading bioarchives right now, start to really look to see work out of Shenyang He's lab and Jonathan Jones' lab and Thorsten Nurnberger's lab. So our whole concepts of PTI and ETI are wrong, okay? So it turns out in order for ETI to work, you need PTI. And so there's an interrelationship here that people are still trying to work out the mechanistic basis of that. So I still think we have a lot of lessons to be learned exactly how we're going to deploy all these various pathways in plants.
Brian, sticking with ROC1, there's a question about, are there multiple alleles of ROC1 that have been compared? And is sequence diversity higher in the ligand binding regions in the LRR?
We haven't, you know, it seems to be we've only found it in the Nicotianas, the ROC1. There's some active alleles, some inactive alleles, and I don't think we've really looked at the binding specificity to see if there are allelic differences there. I could go back and look at that. It's a good question.
Trash day in Berkeley? Sorry.
So I'd like to sneak in my own question here, which is, I was intrigued that when you were talking about that beautiful structure, that at the end of all this stuff you've learned from the structure, a huge amount of learning, you said, and now I have a lot of unanswered questions. I'm just curious what one of your big questions is coming out of this beautiful data. Where do you want to go with it?
Well, you think about TNLs right now. So we know that TNLs, whether it be like the N gene or ROC1 or RPP1, you know, they looks like they oligomerize. NAD somehow, NADase, we don't know whether it's the reduction of NAD or the byproducts of CADPR. And where does that signal? How does that get signaled through EDS and either PAD4 or EDS SAG101? And then further downstream, how is that related to the NRG gene, which is basically probably the executor of resistance in that case? It turns out all these proteins probably have different subcellular locations. You name it, there are so many unanswered questions. I could also give you a little hint, and I'm not a strong believer in doing transient overexpression assays in benthy for cellular localization, but it turns out when you co-express ROC1 and ZOPQ, it looks like it accumulates in the nucleolus. Tell me what's going on there. That's what I mean, okay? Sorry.
A lot of good open unanswered questions there.
Oh, the field is far from over. So for the young students, plenty of stuff to do. Also, I make the other prediction is that hopefully now we'll have a pipeline of making these particular, these various resistosome complexes. And I think it's going to really be important because I think there's going to be various themes that we see. We know with ZAR1, it forms a pentamer, and it's hypothesized that the funnel complex actually inserts into the membrane to actually cause calcium signaling. Again, I think that remains to be shown, but it's a good hypothesis to test. So I think there are many different ways that these systems can work. And especially when we have multiple helper sensors, how are these all working together to give rise to resistance? So the more basic information we get, I'm a firm believer that we can then start to deploy that to do what we all want to do, is to translate this into actually solving real life disease problems.
Right, Brian, one last question from Guido van den Akenbecken. He's looking at
Guido, my man.
He's commenting on your great and diverse ways to protect crops, but you also talked about the challenges. So what is your strategy for implementing this in the field and to get it to consumers?
Well, I think in the case, if we get DMR6 and BS5, and you segregate the Cas9 away in tomato, so since you can actually segregate it away, that at least for APHIS guidelines right now, those would not be regulated as a GMO. So those could actually be implemented in the field right now. So that's pretty exciting. We're also working on, it turns out that tomato actually has a BS2 ortholog that's not active as BS2. So we're trying to do either HDR or gene insertion of the pepper gene into the orthologous gene in tomato. I've talked to the people at APHIS. They said that they don't see any inherent risk in that particular, so that may not be considered to be a GMO also. It's all a matter of what the regulations and what happens in the future and stuff like that. But we have to keep doing the basic research.
All righty. Well, thank you, Brian. That was really interesting and great to hear all your latest data hot off the press. So thank you. And we will move on to, it's my pleasure to introduce our sponsor today, Brian Pusch. And he is the chairman and chairman of the board and the chief executive officer for Microbes Bioscience. Microbes is a biotechnology company located in Woodland, Texas, and it's focusing on developing new technologies for energy and agriculture sectors. We thought this would be really of an interest to our community. The company does many different things, but it specializes in microbial technologies to accelerate bioremediation of contaminated soils and waters and encourage beneficial soil health for increased crop production in agriculture and crop protection. So Brian will share with us the goals of his company and interest to connect with researchers worldwide. Welcome, Brian.
Thank you very much, Mary Beth. And our company is very happy to be the sponsor of the workshop today. And I first learned about IS-MPMI when I was doing some online research related to our business. And I was looking for some information about some of the bacterial strains we work with. And I found a very interesting paper in your journal and learned about the society. And that's when we joined. So I'm an avid reader, and so are others at our company. So I think that, let me share my screen here with what I've got. All right. And then we can go to presentation mode, and there we are. Oops. So, sorry. All right. There's sort of just a list of the topics I thought I would try to cover today about a little bit about our company and what we do. You know, among all the things that Mary Beth mentioned, our focus for today's discussion will be on these things we do in the agriculture sector, which is really the major part of our business. Although we do some other things, as Mary Beth mentioned. So I think in the context of translational science, our company is sort of, along with a lot of others, is one of the participants in the process of moving the research and the research results of people such as those in the audience today into the field. And when we're talking agriculture, it's literally into the field of plants. So I'm kind of hoping that my remarks will give people some insights from a commercial enterprise's perspective on how that process works and how we participate in it. And I think if we look a little bit, here's a little bit of information about our company. We make products that contain species of beneficial soil bacteria and other microorganisms for use in plant agriculture. So we're sort of, if I think about what Jan and Brian were discussing, we're at a much less detailed and less on a macro scale as opposed to a micro scale in terms of the things that we sort of do. Our products are called biostimulants in the agriculture products industry. We have also developed biopesticides for treatment of pathogenic nematodes and fungi. We began in business in 1989, and we've made biostimulant products for more than 25 years. We're sort of one of the earliest companies to do this if you exclude the Thuringiensis, which has been used extensively for decades and has many, many companies selling products in that with that bacteria. Our products work with a consortium of bacteria, so we usually use several strains of bacteria in our products for no particular reason, except it's magic, I suppose. We typically have 7 strains in a product. No, it's really done to try to call on the beneficial properties and beneficial roles of several different species. So, an important part of what we do is to select species of beneficial bacteria that exhibit certain properties or modes of action to produce better plants, higher crop yield, and so forth. So, as is the case for many companies doing what we do, we have strains that assist in nitrogen fixation because of its important for plant growth. We have other strains in one of our products, same product, that has phosphate solubilizers. We also have a third strain that produces indole acetic acid and happens to enhance nitrogen fixation and acts as a phosphate solubilizer, and so on and so on. So there's really quite a lot that you can accomplish with a consortium of bacteria. And typically, if I look at what we understand and what the literature understands about the bacteria we use, you will see anywhere from 3 to 6 known beneficial properties or modes of action in each strain. So when you multiply that by the 7 strains, you're getting what I consider to be pretty good impact on plant growth. We are, because of the way we formulate our products, they are qualified for OMRI listing for use in organic food production. So that also provides plant growers an alternative to some of the chemical products that would not allow their crops to be OMRI listed. And I think if you look around at the ag industry and at the world, there's a lot of concern been expressed about soil health. There's some rather dark predictions about where we're headed with the decline in soil health. I don't know if they're true or not, but I know that it's obviously one of the aspects of food production that troubles people and one that can be addressed. if a grower applies the kinds of products that we produce with beneficial bacteria, they can get better crop yields, and that ultimately can lead to an increase in the world's food supply. Our products and other companies' products like these can be used alongside conventional ag inputs like fertilizer and humic acid and things of that kind. Generally, the work that we've done with field trials and the work that others have done show that these kinds of biostimulants can reduce the need for conventional fertilizer and still increase yields. Some studies have shown you can increase yields 7 to 15% and reduce other ag inputs by as much as 15 or 20%. So those are kind of remarkable results. Of course, they're not for free because people have to buy our products as well. So the growers benefit by getting higher yields and lower costs for their inputs. And generally, you're going to find, I think, that with lower use of other agriculture chemicals, you're going to have a more favorable impact on the environment. So these kinds of products and this kind of use of the underlying science sort of plays into the goals that people have for sustainable agriculture. But one of the things that's discussed beyond sustainable agriculture is regenerative agriculture. And one aspect of that is improving soil health, but as well as other things. And the use of these kinds of beneficial bacteria or other soil microbes like mycorrhizal fungi can be a key element for regenerative agriculture and improved soil health. So I thought, some discussion of how we at our company decide how to make new products and which ones to develop might be interesting in the context of a discussion of translational science. As I said before, we spend a lot of time reading scientific literature to learn about and understand new discoveries relating to plant-microbe interactions. This gives us ideas. Another source of how we decide for a product is through contact with growers and their suppliers. We find out what people are saying, gee, I wish I had, or I have something that doesn't work very well, or plants are developing resistance to something that's been used for a while. Is there something better or different that can be done? And we sort of look to see, do we have some product that we could make that would address those needs? And then we get some ideas from inspiration. We kick around things and say, what if, or is this needed, or is there an opportunity for something that we think can be done? And then we do what I put in quotes here, market research. We find out whether our foolish idea might have some actual commercial value. Our company itself doesn't have an extensive research staff. We do have scientists. So we look to outside researchers for help and ideas. Our strengths are the research at the level we conducted, product development, and product production. And so we're very interested in working with researchers because it improves what we can do and gives us better understanding and better insights into the things we're trying to accomplish. Here, I've just gone through some of the things which I think I've already listed and talked about. Oops, let me go in this direction. So I thought it might be useful to hear some about the process that we go through to develop a product of the kind that we have. We're not a plant breeder, and we are not developing plants. We are developing products that can be used and can improve the performance of both wild type plants and plants that have been extensively breeded to have better traits. As I said, we start with interesting research we've read, or a customer or grower idea or need, or an idea of our own, and we evaluate the commercial viability. We try to understand as best we can the scientific feasibility and the scientific underpinnings of what we think we want to try to do. And then for us, because we work with bacteria, we engage in species selection. We do this based on our extensive knowledge of the kinds of soil bacteria that we're familiar with, and soil bacteria that are in our library that are not in our products, or we spend a lot of time reading the scientific literature. And then we begin by conducting studies in our lab, in our production facility. We evaluate the properties that we hope the bacteria will exhibit, their modes of action, and then we are also looking at the commercial side of things, which is, these all have to be culturable, fermentable bacteria, and we then look at the fermentation yields and how to optimize the fermentation media, which differ from and can differ from species to species. So we're always looking to improve those things and test those out for new products. And because we're putting together a consortium, although it has not been a particularly problematic challenge to us, we do have to test whether, if we pick 3, 5, 7 strains of bacteria to combine in a product, do they get along well with one another? Generally, they do. And then we also recheck the activity of them for the traits or performance we're looking for once they're put into a combination or blended into a product. And from that, we move to sort of small scale, not very scientific, but interesting things of pot trials and greenhouse trials of our own. We do these in our own facility, or we do them in our employees' backyard gardens, or we do them on my farm, just to get a sense of whether we're going in the right direction. And then we figure out what are the next steps that we need to go to. And I think if you look at the things on this slide, if we work really hard, we can do most of this stuff in about a year, but it can take 2 or 3 years if our first tries don't work out as well as we hope. And we may have to modify formulations or substitute species or things of that kind. And then sort of continuing on from that part of product development and introducing a new product, while we have branded products of our own with our company name on them, a lot of what we do is to supply larger agriculture chemical companies with products that they sell either under their brand names or can sell under our name. So in order to do that, we can look to do larger trials in the field, or we can take these products with the data that we have to the larger ag companies. It's obviously more impressive if we can engage university researchers to conduct greenhouse or field trials with our products, or growers as well. If we can gather the data and do the analysis, after the growing season and the harvest, and measuring the yields and evaluating the improvements in the plants and the root system and so forth. But even if we persuade a company to take on a product or product candidate as something that they want to look at seriously, we're still several years away from having a commercial product. Inevitably, any large company, or larger than our company, and our company is not particularly large, will want to do 1 or 2 years of field trials on fairly extensive acreage of our product. So you're looking at a couple of growing seasons at the very least, and hoping that they're as impressed as we are with the performance of the product. And then if it passes muster in the field trials, then there's going to be an entire marketing operation at this ag chemical company or ag products company that will figure out how are they going to introduce the product regionally and geographically, where are they going to go with it, how are they going to promote it to growers, how much product are they going to try to put in the field in the first year on a commercial level, and so forth. So as I say at the bottom here, from the time we at our company are finished with what we want to do, it's still another probably 3 years before there's any meaningful commercial sales of a product.
Brian, we're running out of time, and so perhaps we could close and offer up a question or two before we take a break.
Yeah, maybe just in closing, I guess I thought I would say some of these things that in terms of translating science out, I think discoveries with commercial applications can find their way into the field in different routes, as many of you know. Sometimes it's through ag extension services at the land grant institutions. It's commercialization by companies such as ours. You can start a new company. Other sort of interim roles for researchers to play are conducting the greenhouse and field trials that we've talked about. That's an important part of the translation. The flip side of it is companies such as ours would look to sponsor research at universities. We would look for researchers to be our scientific advisors, consultants to us, and things like that. And of course, there are industry jobs for in the research and development arms of companies. So I hope that's been a useful way of giving our perspective on things, which is at a different part of the overall environment for what we're collectively trying to accomplish.
I think this is a terrific connection for the students who are listening and postdocs thinking about different paths they can go to follow their experience and interest in plant microbe interactions. So terrific. We do have a question that I'd love to get into. Someone asks, are you working on both natural microbes and genetically modified? And if modified, do you need a permit for field application?
Well, we don't work with genetically modified organisms because it presents a problem with organic listing. So, we are more likely to try to find better performing species, but that are naturally occurring ones.
So I have a question, Brian. So you said you're working with like 7 different strains of bacteria, and it seems that most of them are either directly affecting the plant or somehow manipulating the environment to have a different nutrient makeup. What are your thoughts on trying to look at microbes that might produce antibiotics or control the other pathogenic microbes that might be in the environment? And could that cause some problems with these 7 strains that you've worked so hard on?
Well, the truth of the matter is, and I have to be careful how I say this, the Environmental Protection Agency regulates biological products if they are considered biopesticides. And a lot of the properties, you have dual, triple, and quadruple use bacteria that do lots of different things. And in many cases, the sort of nutrient uptake or solubilization, or breakdown of organic matter properties are coupled with what would be biopesticide properties. I mean, if you stop to think about it, all of our antibiotics were derived from soil bacteria. And it wasn't just because they were sitting in the soil waiting for us to discover them for human antibiotics. They're using those properties in the soil. They're using those properties on pathogens or to trigger responses in plants. And so, yes, that's an absolutely important part of what goes on. I mean, some of the bacteria form biofilms, beneficial biofilms on plant roots, just to physically block or make life more difficult for pathogens. And those are some of the properties which I didn't really delve into on things. And, I mentioned that we have a bionematicide product that is an EPA regulated item. It's not yet registered. It will get there. But it's based on one of the species that's in a biostimulant product. A similar thing with the biofungicide that we have. It's, again, species that are also in a biostimulant. So I don't know if that helps in an answer, but yes, I'm very much in favor of that because I think the complexity that's there, as I said to Mary Beth when we were talking the first time I met her, the thing I always keep in mind is that when plants evolved, bacteria already existed and the relationship is there.
Absolutely, very ancient. Well, this has been terrific, Brian. Really great to see that connection, translating discoveries to the field, also looking at how these are actually disseminated. So that's been a wonderful connection there. I think, Mary Beth, do you want to say something about timing for the next session?
Yeah, so we have about 10 after the hour.
Brian Stephenson is a professor and endowed chair of cereal disease resistance at the University of Minnesota. Brian's primary research focus is the study of cereal crops for resistance to rust diseases. with the majority of his work done in barley and wheat. His worldwide collaborations have led to the discovery of important sources of resistance to the UG99 lineage of wheat stem rust, which has been devastating crops across the globe. Brian's research group has significantly contributed to discovering and exploiting disease resistance genes from wild barley relatives for use in modern crop varieties. His group has developed large collections of wild cereal species from across their natural range and is evaluating them for economically important traits. He's a leader for the new International Wild Barley Sequencing Consortium with the goal of whole genome sequencing of 300 accessions from the wild barley diversity collection. In addition, Brian's group is leveraging advanced transgenic technologies to facilitate the rapid and efficient transfer of multiple resistance genes, up to 5 or more at a time, into already adapted cultivars in just 6 months, a process that would normally take 10 to 15 years using conventional breeding. His recent field trial results indicate that exceptional stem rust resistance can be obtained through the stacking of multiple resistance genes into a single variety. These breakthrough studies show that Brian's work is expediting the pace of developing durable resistance in cereal crops, an important contribution to worldwide food security. Today, Brian will talk to us about recent research developments in his talk entitled Busting the Rusts with Resistance Gene Stacks in Wheat.
Thank you very much, Dennis. I really appreciate the introduction. And first off, I would like to thank the organizers of this workshop for inviting me to talk. It's been a wonderful session and I really appreciate it. I also want to point out this is the first panel that I've ever been on where there are 3 speakers by the name of Brian. So that's something extremely unusual. Does everybody see the screen okay? So I'm going to talk today from the perspective of a classical plant pathologist, and most of the molecular work that I'll be talking about was done by Mick Aliff, who's at CSIRO in Canberra, Australia. So just a few words about the importance of wheat. As everybody knows, wheat is the staff of life. It supplies about 20% of the calories consumed by humans worldwide. It is the most widely grown cereal in the world at 225 million hectares, producing around about 735 million metric tons. And within the US, wheat ranks 3rd in terms of acreage and farm gate receipts behind soybeans and maize. And wheat as a commodity is extremely versatile because there are so many food products that can be made from it, from bread to pasta and cakes and noodles. But unfortunately, there are many diseases that can limit production of wheat, and chief among those are the rusts, with stem rust, leaf rust, and stripe rust, all cause very serious losses on wheat across the globe. And in some economic analyses that were done several years ago, it's estimated that the rust diseases cause over $3 billion in losses per year. Now, one of the most important of all plant pathogens, not just the 3 rust, is stem rust, caused by Puccinia graminis form species tritici. This is a pathogen that's really been feared since antiquity as the famine maker. And the reason why is that it can completely destroy crops over a very wide area in just a few weeks of time. Now, under less severe epidemics, we can see marked reductions in yield, kernel size, and quality as well. And if you go back into history, stem rust was really an ancient enemy. The Romans recognized the effects of rust on their crops way back to 700 BCE. Of course, they didn't know what was causing it, but they did know what the effects were in terms of limiting yield. And it was so important to the Romans that on April 25th of every year, they held a festival called the Robigalia to help to appease the rust god Robigus from sending the scourge down onto their crops. And this is very well documented in Roman history. And during the Robigalia, they would march down the Via Claudia and stop at mile marker 5. And then prayers would be chanted, red dogs would be sacrificed, and wine drunk. Well, needless to say, none of this helped mitigate the rust, but it did lead to a nice party for the Romans. But as far as more modern times, stem rust has really been a very formidable adversary as well. It's caused famines and ruined economies for many, many years. And within the United States, we suffered 3 catastrophic epidemics from 1916 to 17, 1935 to 37, and from 1954 to 55. And so we've been through these awful boom and bust cycles so many times with not only the rust diseases, but many other pathogens of cereals. As soon as we find a really nice resistance gene, usually a single resistance gene with major effect, breeders put it into a great cultivar, becomes very popular, farmers like it. And then somewhere lurking, there could be an already virulent pathogen or maybe one that emerges with a new mutation. It can take out the crop, and then we get the epidemic and the complete bust in terms of the acreage that variety is grown over. So we've been through this cycle a few times. I hope we've learned a little bit, and we've had some success with regards to stem rust resistance breeding in the northern Great Plains. That would be the northern part of the US and into Canada. So since that very catastrophic epidemic in the 1950s, most of the hard red spring wheats have been grown, and they have not suffered any serious losses due to stem rust. And that's been really a great success story, and that's still true today. And this is largely due to 3 main factors. Number 1 is the elimination of the alternate host barberry or berberis. This started way back in the 20s. This is the host upon which the rust pathogen undergoes sexual hybridization, so it can generate many new variants that are capable of overcoming various resistance genes, and work way back with E.C. Stakeman and having this large barberry eradication program had been very successful. And now there's really only the last vestiges of barberry and some riparian areas around the upper Midwest, but for the most part, it's been eradicated. The second really important factor, I think, that has contributed to this resistance lasting for a long time is that the USDA Cereal Disease Lab conducts pathogen surveys every year. So they're out looking across the country for potential new virulences that may be able to overcome the varieties of wheat that we're growing with resistance to rust. And then the final factor is really credited to the breeders and pathologists to develop cultivars with multiple resistance genes. And estimates are that there's probably about 6 or more genes in these varieties, and also backstop with some adult plant resistance. So in addition to the success in Canada and the US, there's also been great success with the same strategy in Australia, and also with the Green Revolution wheats that Borlaug bred some years ago. So all of this was fine and good, but we were all sort of a bit in shock when in 1998, William Waigori noted heavy infections on a wheat line carrying the resistance gene SR31 in kind of a remote location in Uganda. And some people doubted the veracity of this discovery, but it was sent to my colleague in South Africa, Zach Pretorius, who in 1999, just a year later, identified it as stem rust, and it was deemed UG99 for Uganda and being described in the year 1999. And it was keyed out to race TTKSK. And the really scary part about the discovery of this new race was that in subsequent tests, it was found to be virulent on 80% and 95% of all wheat and barley cultivars worldwide, respectively. So this wide virulence for those 2 crops is really unprecedented. So it definitely got the attention of many plant pathologists and breeders throughout the world. So UG99 stem rust is it's certainly not static. It spread from Uganda across most of Eastern Africa and made a leapfrog into the Middle East. And the other part that's worth noting is that there are now a dozen variants of the UG99 lineage that are across East Africa and the Middle East. So this has been really a big initiative that's been taken up by many scientists across the globe with regards to the Borlaug Global Rust Initiative. So when we're talking about breeding for durable resistance, we know from experience and theoretical studies that gene pyramiding is going to force the pathogen to acquire virulence, either simultaneous or sequential mutations in the same genotype. And we know that there is strength in numbers. The more resistance genes we can stack or pyramid together, the harder it will be for the pathogen to overcome those resistance genes. Still, I'll never underestimate the stem rust pathogen. I think that the gene pyramiding strategy is likely to be most effective where the pathogen population is pretty stable and also chiefly asexual. And that would be typical of the Great Plains of the US, where we've seen the resistance last for a long time. But I think also one always has to have some backstop major gene resistance along with partial or adult plant resistance as well. So we're very fortunate with regards to wheat that we have many species that are in the primary, secondary, and tertiary gene pools. And so a lot of the work that I've done when I started my position at the University of Minnesota in the year 2000 is that we are taking panels of the progenitor species of wheat and also some of the wheat relatives. And we're putting together panels, diverse panels across the ecogeographic range, and then doing work in terms of identifying new resistance genes for stem rust, leaf rust, and stripe rust in these panels. And as you would guess, there's been many, many genes for rust resistance that have been identified in these wild wheat relatives. It's a virtual treasure trove of resistance genes for us. So it's very exciting to see that we have that ability to really tap into the diversity that we see in wheat and the wheat relatives. And we're doing the same thing also with barley. So I want to I just want to talk a little bit about what the status of introgressing resistance genes into wheat during the time when I first started at Minnesota in the year 2000. We had started working on a wild wheat species called Aegilops sharonensis. It really has its habitat only in the coastal plain of Israel. And nonetheless, it's still quite diverse. And we found a number of resistance genes that are effective against UG99 in the species. And I was working with a colleague at Tel Aviv University, Eitan Millet, and he's a classical cytogeneticist that does wide crossing. And this scheme gives you some sense of what it takes to transfer a single gene from Aegilops sharonensis into wheat. I won't go through all of the protocols, but just suffice to say that there are a number of problems, including low hybridization frequency, poor chromosome pairing, various cytogenetic barriers, and of course, we're also left with a lot of linkage drag, which can be very detrimental. But from this work, Eitan has been very successful in transferring resistance genes, doing the selfing for a number of times, recovering homozygous resistant lines. So then those can then be given to the breeder. And in our case, at the University of Minnesota, that's James Anderson. And this is the normal scheme that Jim uses when he's introgressing resistance genes or other traits into his breeding program. Generally, breeding pipelines taking about 7 to 9 years in order to get a wheat variety that might be suitable for a farmer to grow and that industry would be interested in making bread from. So when you look at Eitan's work over to Jim Anderson's work and finally to when a farmer can get in his hands some of these resistant wheats, the total time from start to finish could be anywhere from 11 to 14 years. So in terms of trying to hasten the development of durably resistant wheats, I think it's an extremely exciting time. There are many genes that are now being cloned quite efficiently from a lot of these wheat species in the different gene pools, and we're actively trying to clone as many as we possibly can with our collaborators. And then I think the exciting research that I'll talk about in just a moment is the ability to now assemble these effective resistance genes into different multi-gene cassettes or stacks. And then with some of the advent of newer protocols for transformation, it's become easier to efficiently transfer these gene cassettes into not only just any wheat variety, but those that are already agronomically superior with high yield, adaptation, and also bread making quality. So it's a big leap in terms of speed from what we were working with a few years back. So some of the exciting things that are happening in terms of resistance gene cloning protocols have sort of evolved over the last few years. And I've worked very closely with my colleague, Bronda Wolf at the John Innes Center. And he's been involved in developing various protocols that can help isolate resistance genes a lot more frequently, or efficiently rather. And these include mutagenesis resistance gene enrichment sequencing, or mute-REN-seq. And there's a variant of this where individual chromosomes are sorted and sequenced called mute-chrome-seq. And then last year, Bronda published on association genetics resistance gene enrichment sequencing, or AgREN-seq. And he was able to clone 2 genes from Agelop's tauschii with that technique in just a matter of about 6 months' time. So we're really going to see a lot of the resistance genes be isolated very quickly. And of course, with all the cheap genome sequencing we have now, I expect that we're going to see many more genes fall very quickly using this protocol. This next slide is maybe a little bit of an extension that Pam Ronald showed earlier. This was given to me by Bronda Wolf, but it just shows from the year of 2014 with the advent of mute-REN-seq, and then how many resistance genes that have been cloned over these 5-year period. So you can see there's just been a really rapid increase in the number of resistance genes that have been cloned for not only the rust and mildew, but some of the other important disease resistance, and also various agronomic traits as well. So it is very much an exciting time to be a plant pathologist. So my collaborator, Mick Aliff at CSIRO in Canberra, was really the lead with Roger Friedman and Diana Horvath in really addressing the UG99 problem using a transgene approach. And so they started this program, and as a proof of concept, they wanted to take 5 genes from wheat and wheat wild relatives, and also 1 from rye. So this was SR22 from Triticum boaticum, which is also wild einkorn, SR35 from the domesticated einkorn of Triticum monococcum, SR45 from Agelop's tauschii, SR50 from rye, and then 1 gene from breadwheat, Triticum aestivum. So all 5 of these genes confer resistance against isolate UG99. And so that was really the main emphasis for this proof of concept. And the first 4 genes listed there are the NBSLR type genes, and they confer all stage resistance. The exception for the last one, SR55, that's an adult plant resistance gene that confers partial resistance. And the unique thing about this one is that it's a multi-pathogen resistance gene. So SR55, it's called for stem rust resistance, but the same gene as LR67 confers leaf rust resistance, the same gene as YR46 confers stripe rust resistance, and the same gene PM46 confers powdery mildew resistance. So Mick and his associate Ming Lu are the molecular magicians that came up with the cassette construction. They just used the basic gateway recombination protocol, and they were able to then construct this 5-gene cassette that is 37 kb in length. And there's just probably going to be next month, I believe, the publication on this work will come out in Nature Biotechnology, so you can read about it at that time and in the next coming issue for that in first of the year. And Mick's group at CSIRO has also a license with the Japan Tobacco Company. So they're using that efficient transformation procedure to transform these wheats. And Mick chose Fielder because it's an easy one to transform, and using the Japan tobacco method, he's been able to get transformation efficiencies of about 50%. Now, we're also hoping that we can get some of these wheats and the gene stacks into CIMMYT, which has as its priority breeding for rust resistance. And so Mick has started some transformations with some of the key CIMMYT wheats. And these include Robin, Kingbird, Mutis, Kachu, Navajoa, and Borlaug 100. The efficiencies at this time haven't been quite as great as with Fielder, ranging from about 1 to 42%. But Mick wanted us to test all of these transformants with the 5 gene cassettes for different rust races that are found across the world that are very diverse and very widely virulent. At the University of Minnesota, we have a biolevel safety 3 containment facility where we can work on such things. And we also have field facilities to also work with transgenic wheat. So when Mick developed these transgenic lines, he had 80 T0 transgenics that were produced. And he found just 5 of the T0s contained all 6 of the PCR junction sequences, and that they were also positive in the DNA blots and the gene-specific probes. And then it was just 3 of those 5, we call P19, P52, and P60, that carried the full intact genes. So from 80 down to 3 that we worked with. And then Mick did an additional step in looking at the T1 progeny segregation, and he did find that the full-length restriction fragments of the 5 genes were co-inherited as a single locus. So this is certainly important for breeders, because if we can have a gene cassette that segregates as a single locus, it certainly makes breeding very simple, that they can just keep that intact and they know that they'll have stem rust resistance. So Mick conducted some transcriptional analyses, and he used RenSeq for this. So this was for the 4 NLR type genes of SR22, 35, 45, and 50. And his results indicated that the transcript frequencies were similar between each of the transgene lines and also the endogenous gene equivalents. So this figure shows the wild wheats with endogenous genes listed. So here's SR35. And then also the multi-transgene lines here. So these reads were mapped to the SR35 ORF. So he's confirming that we have the transcripts that are needed for the resistance. Now, with regards to functional analysis, we don't quite have, with Puccinia graminis form species tritici, all the AVR genes that we would like to isolate. However, there were 2 that were cloned recently, AVR SR35 and AVR SR50. They were cloned just a couple of years ago. And to utilize this information and those constructs, Mick used the luciferase reporter gene, and then he isolated protoplasts from the transgenic lines. And when they were transformed with AVR SR35 and SR50, he found that there was reduced expression compared to the green fluorescent protein normalized ones. And then there was an event that occurred during the construction of these transgenics, and that event was the discovery in Sicily of a new PGT race called TTRTF that had a rare virulence. And unfortunately, it had virulence for several of the genes that we had in our stack, even though the stack was prepared to confer resistance to UG99. The one from Sicily had this unique virulence, but we were able to use this unique virulence to show the function of one of the genes in the stack, SR22. The problem, of course, when you have multiple genes in a stack is determining whether each of them is functional individually. And so by using the AVR genes in the previous slide that I showed, and also this unique virulence about the Sicily race, we were able to show the function of SR22. So this particular race from Sicily is virulent on SR35, and it also is virulent on SR45 and SR50. And then just to separate out the SR55 gene, that's one that is only effective at the adult plant stage. So you're always going to see a susceptible infection types. So you can very clearly see from these large rampant uridinia that these particular lines, both the native ones and the single transgenes, are indeed susceptible to the Sicilian race. But the good news is we were able to show that the SR22 singly and in combination with the other 4 genes gave very strong resistance. And then, so this was a good demonstration, of course, for SR22 being functional. So we were able to show, at least for 4 of the 5 genes, that they were functioning in the gene cassette. So what we were doing in collaborating with Mick Aliff is to really look at a wide diversity of different races with different virulences and really be able to take detailed notes on the resistance reaction. So for the seedling test that we conducted with a number of different races, we used the Stakeman scale, and that's just basically, I'm sure many of you are familiar with, it's just a scale that ranges from 0 to 4, 0 indicating actually complete immunity where there's no visible sign of infection. The FLEC, 0 FLEC reaction indicated with a semicolon. This is where we have a hypersensitive reaction, but absolutely no sporulation of the pathogen. And then the ones have just a small bit of sporulation, 2 a little bit larger sporulation area, and then the 3 and 4 are a much larger and a rampant uridinia forming, and these are more indicative of susceptibility, with the 4 type being completely compatible. So in testing these transgene lines against some of the most virulent races that we have in our collection, as done in the BSL-3 facility, we found that with respect to Fielder, the wild type, it was very susceptible. In fact, near all were complete compatible except to 1 race, and that's one from Ethiopia. Otherwise, all of them gave complete compatibility. So we do know that there are resistance genes in the background of Fielder, but we have plenty of races to overcome that resistance so we can see the effect of the transgene. We had 2 nulls for the 5 transgenics. We didn't have a null for P19, but these nulls that contain only the vector were also showing very highly compatible infection types to some of the races that we challenged them against. And these races represent, as I said, some of the most widely virulent races known in the world. TTKSK here, this is the UG99 lineage that we're speaking about, but we also have some quite high virulence in the USA. There was a race called quadruple TF that was fairly widely virulent, and then a selection from Italy and Ethiopia that and Georgia that round out the group. But what I want to focus on here are the results for the 5 transgenic lines. And you can see that across all of those very widely virulent races, that most of them are giving immune or near immune reactions. There were just a few cases where we saw very, very minute sporulation. In fact, they were given a 1-, which is a Rust notation for a smaller than normal sporulation of a 1-type pustule. The other thing we wanted to do is to make sure that these transgenic lines were effective in the field. And so we established a transgenic testing site just south of the Twin Cities in Rosemont, Minnesota. We tested these lines in 2018 and 2019. What was very critical for this test is to make sure that we found a race that was originally identified from Minnesota that would be virulent on Fielder, so we could detect the transgene. So there were a couple available, but certainly we're not able to put out some of the most widely virulent races that have been found in the US. We have to keep to the Minnesota origin races. So all of this material was planted by hand. It's much easier than doing anything with a machine. And then in this case, we really wanted to challenge these transgenes. So we inoculated 4 times with very high levels of infection, very high levels of inoculum. And then we read the plants, and I had my colleagues from the Serial Disease Lab come by for when we were doing the Rust scoring so they could see the results of the experiment as well. So again, we wanted to take very detailed notes on the level of resistance conferred in these transgenic lines. And so we normally rate based on 2 criteria. One is the rust severity, that is the percentage of rust from 0 to 100 that are occurring on the stems. And then we also look at infection responses. So they're the individual infection types. And with an R-type reaction, we see just a very minute sporulation area. And then with an MR, a little bit larger sporulation area. And then MS, much greater erumpent pustules forming. And finally, the susceptible are completely compatible. But for this particular study, we established one more criteria, and that's an HR, or highly resistant category. And that's when you can either see flex or see nothing, but absolutely no sporulation is occurring. So in our study, then, we planted out the transgenic lines in multiple replications, included the controls of Morocco, planted on 2 planting dates. And you can see from the top 5 categories here of accessions, Morocco ranged from 65 to 100% severity and all had susceptible infection types. The Fielder wild type was almost as bad in terms of susceptibility at 95% severity. And then we had the 2 nulls that were available also having quite high severities and susceptible infection types. But what was really remarkable for us was how strongly resistant the transgenics were. I spent probably 5 or 10 minutes rating each accession just to look to see if there was one sporulating pustule, and there was none. It was absolutely resistant, very highly resistant. So they were given a severity of 0 and then highly resistant infection types. And you can see just the results of these 2 stems here of the level of rust on Fielder versus what we call the big 5 with the stem rust resistance genes of the 5 genes together in the background of Fielder. Now, I mentioned that one of the genes that was put into the cassette was a multi-pathogen resistance gene, and that is the SR55/LR67, YR46, and PM46. So we're also interested in leaf rust resistance. So in 2019, we established a leaf rust screening nursery for the transgenic lines. And we established the epidemic a little bit differently. In the first case with stem rust, we applied the inoculum directly because we wanted to really give it a hard test. But when we're dealing with leaf rust and a partial resistance gene, we inoculated susceptible spreader rows and allowed the inoculum to spread naturally into the test entries. And if you take a look at the data here, again, Morocco was our susceptible check once again and had very high severities, whether it was planted early or a week later. And they range from 80 to 85% with susceptible infection responses. Fielder is not quite as susceptible as Morocco at about 50% severity. And then I just want to draw your attention down to the transgenics. So they had severities ranging from 5.8 to 16.3. And they had quite often the moderately resistant to moderately susceptible infection types. So all of these were much lower and statistically lower than the Fielder wild type. And then as additional controls, just to prove that LR67 was the gene responsible, these are the single gene transgene lines with LR67, and you can see the level of leaf rust severity that they had. Now, one perplexing thing to us was that the two nulls actually had quite low leaf rust severities, and we're not quite sure why. I've talked to Mick about this to try to explore why this might be the case, but that still needs to be resolved. And just to give you some idea about the differences in infection, this is fielder, a little bit late in the season, but you can see the leaf rust occurring and pretty high severity across that leaf versus the other big 5 transgenes, which had extremely low severities on them. So this LR67 providing really a nice level of partial adult plant resistance. Now, we of course also were interested in seeing if this gene cassette could confer resistance against the wheat stem rust trifecta, stem rust, leaf rust, and stripe rust. So we tried to establish a stripe rust nursery in the year 2020. And unfortunately, COVID got us literally. We weren't able to get our nursery established because of all the difficulties in getting back onto campus for essential employees. So unfortunately, that will have to wait until next year. So just to summarize, this 5 stem rust resistance gene cassette was assembled and transformed into cultivar fielder. This cassette was integrated as a single locus, and the functional analysis was completed for 4 of the 5 genes. The one missing one was for SR45. Now, in our seedling assays against a very wide and diverse panel of isolates, the 5 gene transgene lines were very highly resistant, including to those in the UG99 lineage. Of course, the Sicilian race has us very concerned. It's a wake-up call for us to make sure that whenever we develop new stacking cassettes, that we make sure that they're tested and vetted against a wider panel. But as I said, in this proof of concept, we are really focused just on resistance to the UG99 lineage races. Then in the field test, these lines performed extremely well. All of them gave near immune reactions to race QT-HJC as adult plants in the field. As far as the LR67 multi-pathogen resistance gene, as I said, that confers a non-hypersensitive type of resistance. So it's a partial resistance that's manifested only at the adult plant stage. And those 5 gene transgene lines had anywhere from 37 to 79% lower leaf rust severities than the fielder wild type. Very, very encouraging. And we hope that we can get tests going against stripe rust and powdery mildew to test the multi-pathogen resistance gene in 2021. So the really exciting thing for me, since the time when I first started my job and the very laborious method of transferring a single gene into a wheat line, is the ability now to transfer a 5 gene cassette to possibly 4 important diseases of wheat, and to put it into a wheat cultivar that would be already acceptable by the public and the growers and the industry. So it's very much a quantum leap over where we were about 2 decades ago when I started my job. So I'm sure there's a question about the cost of fitness with regards to inserting this cassette into wheat. And what we did to try to look at this was to take our homozygous transgene lines and compare them with the respective null segregants and also the fielder wild type. We did this all in the greenhouse, and we looked at growth, development, and yield parameters such as heading date, height, tiller number, leaf size, seed number, seed size, and seed weight. And fortunately, we didn't find any significant phenotypic differences between the multi-gene transgenes and the control line. So really no clear pleiotropic effect arising from the transgene expression. So I think I'm just to the maybe the slides is not advancing right now. Not sure if you can, on your end, Nelson, if it's possible to advance the slide. I know we're just right out of time here, but yeah, I'm not able to advance the slide just now unless you're able to, Nelson.
Brian, were you at the end for acknowledgements or did you have more content?
Actually, I think I just had 1 or 2 slides left. I'm not sure if...
Nelson, maybe stop sharing and then reshare to see if we can solve that.
Maybe we could try that, yeah. If not, I can just talk through the last ones very briefly and show the acknowledgement just by... I may be able to do it, Nelson, no?
I just
Okay.
advanced.
Okay, yep, okay, thank you. So just as future ongoing work, we're continuing to pursue our gene cloning projects with some of the different wheat relatives. And given the situation that happened with the Sicilian race, we are going to really robustly test each resistance gene to make sure that they have the wide degree of effectiveness that we need in a cassette. And we're also very cognizant of the fact that we need to practice gene stewardship. We don't want to have any of the cloned resistance genes ever be deployed singly. It's not a good idea. And also, we want to continue to build bigger and better cassettes that may lead to the development of more durably resistant wheats, eliminating, I hope, the threat of rust diseases. And if you could advance one more slide there, please, Nelson.
Give me a second. Hold on. Let me stop and start it again.
Oh, sorry. So some of the interesting news is from Mick Aliff's group just recently is that he's able to build bigger and better cassettes. He now has online what he calls a Big 6, a Big 7, and also a Big 8 gene cassette. And this last one is very interesting because it contains several adult plant resistance genes for both stem rust and stripe rust, and plus has the LR67 gene for leaf rust resistance. So we're anxious to see how this one will perform in the field. And if you could advance the next slide, if it's possible. I just wanted to make just a couple of comments about GM wheat. Is it a possibility? I mean, we have this incredible technology now to develop, we hope, durably resistant wheats to the rust. I understand it's a very long and expensive deregulation path. There's still very strong public opposition to this technology. And really, as indicated in the article in Science a few years back by Wolf and Duga, that there's really a lack of interest in major biotechnology companies to really take this forward. So it may be that we're going to have philanthropic organizations fill this void. And I hope that also in the future that maybe gene editing can accomplish the same goal and give us greater acceptance of GM wheat. And I just have the acknowledgements on the last 2 slides, Nelson, if you can help. So I worked a little bit with Norman Borlaug in the last years of his life with the Borlaug Global Rust Initiative. And it's a very much appropriate time right now that it's been 50 years this coming December 10th that Borlaug received the Nobel Peace Prize for providing the wheat that provided the bread that made peace in the world. And I know that Norman Borlaug was always accepting of new technology if he thought it was going to be effective and safe. And I know that he would have taken this technology under his wing and promoted it as strongly as he possibly could. Next slide, please. So I just want to thank very briefly the funding agencies. I'd like to particularly state the funding by the Two Blades organization with Roger Friedman and Diana Horvath. And then the last slide, I want to just give all the credit I can to my cooperators who've been so wonderful, and they're really the ones who have pushed this this technology, and I just happen to be the lucky plant pathologist that gets to test these in the field, which is really fun. So with that, I thank you very much for your attention, and I'll try to answer any questions that there might be. So thank you very much.
Thank you, Brian. That was really exciting talk, and it was great to see all that work being done. I was commenting to myself several times during your talk about the parallels that I see between the stem rust work that you and the community have been doing with our work in potato and the community looking at resistance to Phytophthora. And I guess we can fight over which one we should call the famine maker pathogen. But using wild species to identify resistance, using RenSeq and trying to stack genes for resistance, it's really a similar approach that people have been using to look at Phytophthora resistance in potato. We'll see how it goes.
Yeah,
so
I've been a good follower of Mark Gislane's work and deploying those resistant potatoes in Africa. It's wonderful work, and it's exciting to see how well it's done.
Yeah, and David Douches has a USAID project with Bangladesh and Indonesia also to release a different 3-gene stack in those places in potato. So
yeah, that's very promising.
I have a question to start out really quick. So as far as releasing these 5-gene stacks, or 6 or 8, or I look forward to the big 10 stack at some point. Do you have any idea what the stability of those genes might be? Not in the respect to being overcome by the pathogen, but just their genetic stability. You're stacking together groups of 5 to 10 relatively similar genes in their genetic structure. Is there a chance or a worry that there might be some recombination to knock out certain genes over time?
Yeah, that's a very good question. I don't think we're quite to the stage where Mick has looked at that yet, but we definitely will be paying attention to that. I know that as he gets the larger cassettes, that you start to get partial integrations and multiple loci integrations. And so we have to look through that. So he's trying to, identify the cleanest lines, for testing, so we can really see what the effect of each of the, single locus transgene cassettes are working.
Right. So
wonderful talk. Should I
Jean.
take the next one there, Dennis?
Go ahead, Jean. Yeah.
So great talk, Brian. I was just going to ask the next one, which is, as an alternative to pyramiding multiple genes, could a single strong resistance gene that recognizes multiple alleles of a widely conserved effector lead to stable resistance as well?
It's possible. A lot of my early work on durable resistance was with barley, and there was a gene called RPG1, and we thought it was pretty invincible because it provided durable resistance from the 1940s all the way up into the 1990s. But then guess what happened? A virulent race arose in Canada, we believe, and it may well have come from barberry. And that overcame that resistance gene. So we know that gene is vulnerable. We had, great hope for it. I still think it's a useful gene because it is widely resistant to many races, but I would love to be able to identify a gene like that. But I mean, I'm optimistic that maybe we'll find other things like the SR55, LR67 type 1 that are multi-pathogen resistance genes. That would be wonderful to have additional genes like that, because we need the adult plant resistance conferring partial resistance to backstop any of those major genes, which I still think are prone to be overcome. With the example of the Sicilian race, here we were trying to form the cassette against UG99, and then somebody identifies the Sicilian race, which overcomes, 3 of our 4 NBSLR genes in the stack. So a red flag warning for us.
Right. So a question from down I-35 from Roger Wise. He points out that SR35 is an MLA ortholog, and I think you mentioned that you're planning on doing stripe rust and powdery mildew resistance screening at some point. Are you planning to test a wide variety of strains of powdery mildew and stripe rust?
Well, we're not quite as well set up to do mildew work as Roger. So we usually just do really low tech. We usually just see what isolate is cropping up at the greenhouse and we'll test it to that. So we might have to get in touch with some of the folks that do wheat powdery mildew work on a routine basis. I know Roger does it for barley, but we may have to ship these out to some other folks to have them tested. We're all set up to do the rust. We have big collections of all three rusts and the BL3, but the mildew we may have to put in the hands of some other folks.
Ailing Jin says, great talk, and asks whether you have observed any transcriptional gene silencing of the stacked transgenes when they contain homologous domains, kind of related to Dennis's question.
Oh, you know, I'm not sure if Mick has done this yet. I asked Mick to see if he could be on this workshop, but he said, "Oh, it's 3 in the morning for me, no chance." But that one I may have to defer to Mick. But I don't think he's done that yet. We haven't got to analyze all of those yet.
So we have a question about the logistics of releasing these varieties once you get approval and all that. So you mentioned that it would be important to not release single gene varieties at that time. Do you have any kind of idea how you might prevent companies, commercial breeders, from releasing varieties that have single gene resistance?
Yeah, so with Roger Friedman and Diana Horvath with the Two Blades organization, they're trying to protect the genes that are cloned, some of the work that they're funding. So that's probably how we can best keep those genes from being deployed singly, I guess, is to have the IP protection and then to only put them together as gene stacks.
Well, that would be for transgenics, but I think, you know, through conventional breeding, could breeders potentially release those genes?
Yeah, they probably could. Yeah.
Okay. All right. I mean, it might be, I was thinking, you know, would you plan to release kind of intentionally single genes in small amounts to test whether or not the strains that are out there are actually overcoming genes that are in your stack, to give you a better idea of when those 5 genes might be overcome?
Yeah, I mean, that would be, yeah, you know, the USDA Cereal Disease Lab has these trap plots, you know, throughout the country. And so they have many of these single genes. I don't think they have all the new genes that we have. And of course, some of them are in wild species, which just don't grow well. And actually, they may be considered noxious weeds. So we've got to get them into some background of wheat to check them. But for now, we do all that in the seedling greenhouse and just take all the races that are potentially different and evaluate them at the seedling stage. So that's how we do it now. But I mean, the whole thing with the Sicilian race, I mean, you know, that was really a one-off thing, I hope. But, you know, I really don't trust stem rust. I mean, especially in areas where there's still sexual hybridization going on. There's some places in Spain and other places in Washington State and British Columbia. And one of the things we're going to do with Dr. Yu Jin and Pablo Oliveira, my colleagues at the Cereal Disease Lab, is we're going to bulk take a composite of sexual population from barberry, and we're just going to throw it on each of these transgene lines to see if anything pops up that could potentially overcome it. So that has 2 purposes. One, it'll inform us about what genes might be good or bad, but it also may help us in terms of identifying the function of some of these individual genes in the cassette.
So we have a question from Nicole D'Onofrio, who says, you mentioned barley a couple of times. Are you doing parallel experiments in barley against rust and other fungi?
No, unfortunately, barley is one of my favorite crops. It's just kind of an orphan. So we have found a number of other genes that we've put in conventionally, and they have all been cloned, RPG1, RPG4, 5 by my colleague, Robert Brueggemann. But we just don't have the funding and wherewithal to do it for barley. Having said that, there was a couple of genes recently cloned from wheat by Bronda Wolf, and they put those into barley as just an easier transformation vector to study, and they worked really well in barley. So unfortunately, just due to barley not being a really worldwide important crop, except for beer and whiskey and so forth, we just don't have the effort put into that, what I would call an orphan crop.
Right, we have an interesting and important question about IP protection. So you mentioned that Two Blades would working towards getting protection of the genes that you have in the stack, and there's some concern over being or making those genes or this stack available to places in the world that would really need it, and what costs might be associated with that. I know some intellectual protection is more just to kind of control its release and not necessarily make a whole lot of money. What's the philosophy with trying to roll out these gene stacks?
Yeah, it's Two Blades philosophy that this should be shared. This technology should be shared. So they're working very closely with CIMMYT to get these into the CIMMYT wheats and to have CIMMYT test them. And so these discussions are ongoing right now. And at the same time, we're also trying to get some of these field tests put into Africa in the places where there's hotspots for the UG99 lineage races. So yes, the Two Blades organization definitely wants to get this into the hands of organizations that will need the help and the subsistence farmers. And I guess the charge would be for commercial wheat breeding operations that use that IP.
Great. So I have one more question. I think we've answered most of the things in the Q&A. So I guess this may not be an answer to this. So by releasing these gene stacks, you're putting basically extreme pressure on the pathogen to overcome that resistance. Do you have any strategy in place to maybe plant bait line something to keep the pathogens from having to evolve to overcome these genes?
Yeah, that's a great question. And when people started talking about putting these multi-gene cassettes together, I mean, I think back to, the diversification of the crops, like, multi-lines. And I actually had the idea that if we have enough of these stacks, we could potentially try to deploy different gene stacks in different areas after being informed about the virulence. And one could maybe even try to do some of the repeat of some of the multi-line work that was done in some of the oat crop rust studies in Iowa a while back. And in this case, you'd have it with transgene technology. So I would opt for diversification of the gene cassettes in addition to, the gene stacks, but also to make sure that we have an adequate level of adult plant resistance backing up those major genes. Because like I said, I don't trust stem rust. It's very naive to think that we can control that indefinitely in the future, especially if it's a sexual population coming off a barberry.
We feel the same way about Phytophthora.
Yeah, definitely.
Well, thank you, Brian. That was a wonderful talk, and we can pass it over to Mary Beth to wrap things up.
Thank you.
Thanks, Brian. I'd like to take a moment to thank all of our speakers for their inspiring and intriguing presentations and ideas for moving us forward on our research. And I'd like to also thank the attendees for giving us great questions and probing the content for today's discussion. I'd also like to invite you to join us in our next research webinar on translational science, which will be held in February 2021. Is that slide popped up?
Yes.
Yes.
Okay, great. Well, we have another wonderful panel, stellar scientists, including Bing Yang at the Danforth Science Center. Hailing Jin at the University of California, Riverside, Carolyn Young at the Nobel Research Institute, and Nick Runwald at Oregon State. And so they're going to give us some new different topics and their advances in translating their research to the field. Finally, since we are all remote and in the virtual space, we plan as a society, IS-MPMI, to continue holding quarterly meetings throughout 2021. As you know, we don't have our annual meeting this upcoming year, and so it's an opportunity for us to stay connected, hear the latest updates from our colleagues, and bring you the opportunity to continue in the dialogue of our community. So I encourage you to please send suggestions of other topics that you'd like to participate in. I put my email in the chat. You can send, did that go through? Send your ideas to my last name, mudgett@stanford.edu, so that we can get your input and line up some more speakers. We have a list already, but we want to hear from you first before we move forward. And then lastly, I'd just like to say it was great to see so many of our colleagues in the attendees list. Hope you're doing well, and our meeting is now closed, and we look forward to seeing you again. Bye-bye.
Thanks.